Good morning, Deepak.
Very good morning.
Good morning, everyone.
Morning, Ravi.
Good morning.
So very good morning to all of you and welcome to APA India 2021 virtual conference. Today's webinar 18, the concluding webinar, the last webinar in a series of 18 webinars. So would like to welcome all the participants and we have still 4 minutes to go. So at 8:30 we will get started. So till then be there, keep your lines muted, and we are already live on YouTube as well. So just be there, another 4 minutes, we'll get started. Thank you. All right, so very good morning to all of you. And before I start, let me just quickly confirm. Purushottam, can you hear me clear?
Yes, Deepak, I can hear you very clear.
All right, thank you. Thanks a lot. So very warm welcome to all our participants. A very good morning to you on a Saturday morning. Today is webinar number 18, final concluding webinar in a series of 18 webinars as a part of APA India 2021 virtual conference. So the monthly theme for this month of November is emerging challenges and way forward with small molecule drug development. Today's session is chemistry, manufacturing and control. So today's CMC session in the last webinar, not only for CMC, but also for the entire APA conference. Today is the last webinar. So we are going to conclude the conference today. So I think it's my duty and would like to take a few minutes of yours and take you through these slides, especially to express my gratitude and thanks to our organizers. Because friends, this has been a very exhaustive webinar series of almost 18 webinars, 6 months, 3 Saturdays every month, starting from June till November. So the credit goes to all our organizers for their continued effort and their active participation. And it is because of them, we could actually put up such an interesting agenda on board. So a special thanks to the presiding chair and chair-elect. And in fact, today I will take the names also so that it really, it is worth recognizing their efforts. So presiding chair Anurag Sood, chair-elect Ravi Trivedi, for regulated bioanalysis, presiding chair Amarnath Jaiswal, Chair-elect Shefali Gupta. For Discovery, Chair Ravi Kumar Trivedi and Chair-elect was Jignesh Kotecha. For Biotransformation, Prakash Vachaspati and Chair-elect was Samula. And Chemistry, Manufacturing and Control, Rajesh and Purushottam Signurkar were the Chair-elect. So a big thank you to the presiding chair and Chair-elect for chairing the committees. And the Chair-elect will resume their role from today onwards. They'll be the presiding chair for AP India 22 for next year. So as I always mention that chair-elect and presiding chair, they are supported by a very active organizing committee team. And mind you, all these people are really busy. And they, as you can see, they represent more than 40 different CROs and research companies and academia operating out of our country. And they have a day job. And in spite of that, they are committed to AP India chapter. So I would like to take this moment to thank each one of them for their contribution and for their continual support to make AP India chapter successful since last 12 years. So at Regulated Bioanalysis, the committee comprises of Darshana Jani, Srinivas Savale, Patanjali Kadiyala, Venu Madhav, Peter Van Amsterdam, and Aman Dikshit. For Discovery, Jonathan Joseph, Prashant Kole, Vijay Rana, Bhavesh Patel, Ravi Shankar, and Shrikant Dittakavi. So a big thank you to all our very active committee members. Again, for Biotransformation, Rama Iyer, Kaushik Mitra, Selvan Ravindran, Ujjal Sarkar, Mohammad Zainuddin, Zakir Pinjari, and Vinod from Laksai. So a big thank you to the committee of Biotransformation. And finally, Chemistry, Manufacturing and Control, Sailesh Waria, Hugh Hughes, Satish Singh, Anurag Sood, Pankaj Shah, Hemant Bhutani, Ajit Narang, Vishal Nashin. So big thank you to the entire committee of CMC as well. So thank you to all the organizers for their great effort and commitment to make AP India 2021 virtual conference a successful conference. So a big thank you to all of you. Again, without the support from sponsors, it's just not possible to run such an international meeting. So big thank you to Shimadzu, SciEx, ZEFSci, Phenomenix, Thermo Fisher Scientific, Agilent, Syngene, USP, NATCO, Vida Clinical Research, Bristol Myers Squibb, Zoetis, Nanotemper, Invitro Admet Labs, Origin, Vuxi Aptac, and Loris Infosystem. So a big thank you to all our sponsors, and would like to state that many of the sponsors, they've been associated with AP India chapter since last decade or so. So a big thank you for their continual support, and I'm sure in future also we'll keep working together, and hopefully we will have your support for AP India 2022 also, which is most likely is going to be an in-person meeting. So big thank you to all our sponsors. And this year we collaborated with Life Sciences Sector Skill Development Council, which comes under Ministry of Skill Development and Entrepreneurship. And through that, we got access to the entire academia, all the pharmacy colleges and even the science colleges across country comes under this council, which is under Ministry of Skill Development and Entrepreneurship, Government of India. So we are thankful to Life Science Sector Skill Development Council also And in future also, we'll continue the collaboration with them so that we can reach out to the academia also as effectively as we are reaching out to the industry. So big thank you to LSSDC. And just a couple of now announcements for the Kindle prizes. Like, as you know, in every webinar, we used to do that. So I'm glad to share that finally the Amazon have actually confirmed. As you can see here, a lot of names are there. The ones which are in the blue and highlighted in yellow, I think they have already received, I got the confirmation email from them. They have received the gift card. So would request all of them to please check your emails. In case if you haven't, then please come back to us. And the ones which are there in the red, I think most of them are from Syngene International. So I would request all those participants to please confirm once if it is okay to send those email, because that email comes from Amazon directly. So if your server would allow the email at your official email, only then you will be able to access it. So in case if you want to share your personal email, please do so. Otherwise, by Monday, those will be released on your official email IDs. So if the server doesn't allow, then you will miss it. So please come back to me on that. And by Monday, hopefully, a few which are remaining will also be released. But don't worry, if at all if you have any issues or if you haven't received your gift card, please come back to us and we will definitely help you out. For the poster session, we had a very interesting poster session on 13th of November. There were 45 posters in total. And out of that, 9 posters were selected. I mean, they are the winners. We have 3 first prize, 3 second prize, and 3 third prize winners out of that, which we are going to announce today after the webinar, after the talks. And this year, we have started a new initiative to encourage academia participation and their research. So it's Professor Saranjit Singh Award, which will be given to an outstanding poster presentation done by an academia or academic student or a faculty. So that winner would get a trophy, free registration for next year in-person conference, to and fro economy airfare to the venue from wherever they are residing in country, and 3 nights accommodation at the conference venue. So these are all the prizes for the poster winners today. So I'm sure all the poster presenters must be excited. So we would like to wish them all good luck to you, and just hang on there till we announce the winners. Finally, I think it's time and we can get started. So for Q&A, while the talks are going on, as usual, if you have any questions, please post your questions on Q&A tab. You can also put it on a chat box, and we'll try to answer your queries during the webinar itself. Suppose it's not possible because of the time constraint, then do write to us at bostondb21gmail.com, and we will get your queries clarified from the concerned speaker. This is the last webinar, and even today, at the end of the entire webinar, we will have this Kindle draw. So there are a couple of very surprise gifts for today, especially for today's draw. So please be there, attend all the talks, the concluding session, and be there till the end. We'll be doing at the end of the webinar this draw. Finally, after today's webinar, hopefully in next week, we will be able to upload all the presentations, all the webinars on our YouTube channel. So we'll be sharing the link with all the participants, and feel free to go back and refer any of them based on your interest level. So with that, I think we should get started. So it's time. So let me now invite, without further ado, let me invite the moderator for the session, Dr. Purushottam from Syngene. Over to you, Purushottam. Thank you.
Yeah, thanks, Deepak. Good morning, all. I know it's too early morning for many of us, right? But it's very important session today. We have quite a very interesting topics lined up for today's webinar, especially in the area of emerging challenges and way forward with small molecule drug development. And of course, these are actually challenges where each and every industry runs through every project, and it's very important learning. We have excellent speakers on board today to share their learning and experience in the area of multi-parameter stability characterization, pattern crystallization process and development, and the most important part is also how do we bridge the technology advances with regulated bioanalysis. With that, I would like to introduce our first guest speaker, Dr. Saji Menon. She is an application scientist at Nanotemper Technologies, where she handles the application and support on bridging protein characterization tools to the customers in India. Since joining Nanotemper in 2016, she has been providing application-based support to all the existing users of Nanotemper technologies and to new customers, helping them with all matters related to technology and assay development. Prior to joining Nanotemper, Saji did her research in the field of plant molecular biology at very well-known institute, what I would say, Birla Institute of Science Research, Jaipur, and was involved in plant morphological, molecular, and genome-based studies. She was also involved in understanding the molecular diversity of medicinal plants and applications of drugs. Very interesting talk today. We have really understanding the multi-parameter stability characterization. And I would like to welcome Dr. Saji for her speech.
Thank you, Dr. Purushottam. I'll just share my screen. I hope my slide is visible.
Yeah, we can see that.
Okay, fine. Thank you, APA, and thank you, Dr. Purushottam, to introduce me. Today, I'm representing Nanotemper Technologies, and I'm an application scientist for the company, and I take care of the Indian region. The company basically has a portfolio which is into, product characterization, basically, which helps in the early drug discovery and late drug discovery phases. So today's talk is revolved around the equipment called Prometheus Panta, and my talk is called multi-parameter stability characterization of biologics in formulation and development. Before digging deep into exactly what this technology is and where we can use it, I just want to give a brief overview about the company. The company started in a small garage in LMU, and the basic concept of the company was revolving around the fast thermo-optical particle characterization of molecules. And this actually was a patented technology by the two CEOs of our company, who during their PhD was working on the particle characterization. Based on this technology, they developed the first equipment in 2010, which is called Monolith NT115. And the main aim of this equipment was to understand the binding interactions of two biomolecules. This was widely used. People started recognizing it, and they started using it in their various workflows where two biomolecules can be easily characterized. And with subsequent usage and constant support from the customers, the users, we developed the automated version. But on the sidewise, we also developed a very interesting equipment, which is Prometheus NT48, that was basically designed to understand the stability parameter of a protein molecule. This instrument was also kind of used widely, and with the subsequent usage, we developed the automated versions. And recently in 2020, during the COVID phase, we developed the new version of the same equipment, which is known as Prometheus Panta, of which the talk is today. And the interesting feature here is that we are not just understanding the TM melt of the protein, but we are also understanding the aggregation and the size of the particular aggregate. Now let me go what exactly this equipment is about. So we all know what biologics are, and why they are important, because in today's challenging field, and especially when there are a lot of disease, for example, the COVID that has been widely spread, we need to have more and more therapeutic drugs in the market. And each and every biopharma or the pharmaceutical company is actually devoted towards producing a lot of drugs, and they are actually doing a lot of work to keep this pipeline coming in. So as we know, there is a very healthy pipeline, and to get this pipeline approved, really rigorous efforts are put. So we know if there are 1,000 molecules in the market, it's really challenging to put all the 1,000 molecules into market and then commercialize it. So for these, lot of approvals, lot of study is required. So that's where we bring something interesting, which is called stability characterization, and that goes like a workflow in our early discovery and late discovery stages. But there are very big challenges when you look into a stability parameter. It's not just that we are studying TM of a particular protein. When we look into the protein molecule or the complexity of its protein, there are different challenges, because protein in its native form, or it in a different buffer or in different environmental conditions, how it behaves, we never understand. So there are various challenges when we are studying the biologics in a different way. First is the complex stability matrix. The second is when they are unfolding, we should understand how they are unfolding. You need to understand the different transitions when we are understanding the stability parameter. To understand all these things, we need to really have a high throughput. So all these things are actually going hand in hand when we are designing a drug, or when we are actually putting a lot of efforts to make it available into a market product. So when we talk about stability, we have a lot of attributes around it. It's self-interaction, cross-interaction. We need to understand the colloidal stability. We need to understand the chemical stability, or we need to understand the thermal stability. So these are like certain attributes that revolve around stability. And there are various high throughput equipments which are designed to understand each of these parameters separately. Now, though we have so many technologies, we have something in mind that when we look into a stability data, what should be there? I mean, what should be the background? First and foremost, we need to understand the resolution should be very good between two different TMs, or if they are similar domains or there are two different domains, the resolution of those domains should be very precise. Then the sensitivity also matters, because signals, if you identify different domains, you should have definite signals. Another important aspect we look into it is the concentration, because if we are limited with concentration, detection won't be happening. So we always think that we should take lower concentration or higher concentration. The range should be there. Then the specificity, in a way that whatever buffer you're using, the instrument should be compatible. Another important aspect we look into is precision and accuracy, because if we are analyzing one product one day, the analysis should be almost matching when you're actually analyzing it after a period of time. So these kind of parameters are always there in the background when you really want to understand the stability data. So here we have the product line which we will be addressing today. So as I told you, Prometheus 48 was the first one which came into existence. And then with the broad usage of all the equipments, we bought the automated, and recently we introduced the Prometheus Panda, where we are actually not confined ourselves with the thermal unfolding profiles, but we are also understanding the aggregation as well as the particle size, I mean the size of those aggregates which are formed. Now I will tell you how the technology works. It's a very simple concept. It's a very tabletop equipment where we have two detectors. One is for understanding the TMs, which have the fluorescence and the back reflection LED and detector. And the other one is a DLS unit, which has DLS laser and DLS detector. And these optics are fixed, and we have a thermal plate on which we align the capillaries. So the mode of measurement is by using capillaries. Each capillary, they are glass capillaries, and they intake almost 10 microliter of the sample. The sample concentration range is also there. We take 5 microgram to 250 mg per ml. So that's a broad range where we can analyze our samples. And the thermal ramp that thermal element takes a temperature from 15 to 110. So it's a broad temperature range we can measure with. And what we are doing here is the fluorescence and the back reflection LEDs and the laser, they actually understand the thermal unfolding of the molecule of interest. And if there are any aggregates during the analysis, we can side by understand the size of the aggregates also with the DLS detector and laser. So this is how the whole system is designed. Now, let me go back how the technology works. So as I told you, there are three important parameters here: thermal melt, aggregation, and DLS data. So what exactly we are measuring, we are understanding TM is by understanding the intrinsic fluorescence. So any molecule, that is the protein molecule, in its native form, have its tyrosines and tryptophans barrettes, which gives the maximum intrinsic fluorescence. So when you start to give a temperature to it, it tries to unfold. And because of its unfolding formation, the tryptophans or the tyrosines that are buried inside, they try to open up and give the fluorescence. So you can see a data that we count from folded to the unfolded nature, and where we conclude the TM or the chemical stability of the molecule is when the molecule is in a half folded, half unfolded state. And we get the TM data. So this is the first data that we record. Next is the aggregation data. So how the aggregation is calculated here is by a back reflection technology. So what is actually happening here is when you are passing a laser through the surface of the capillary, inside the sample, if you see aggregates, the back reflected light will always be scattered if there are aggregates. If there are no aggregates, the whatever path of the light is in, it will almost go in the similar format out. So if there is an aggregation, we calculate the onset of that is the temperature from where the aggregation starts. So this is how the back reflection technology works. And now we need to understand the size of the aggregate. So we have introduced the DLS part here. So we all know how DLS works. When you put a laser to it, the motion of the molecules will give you intensity scattering, and that intensity is actually calculated as an autocorrelation function. And based on this autocorrelation function, you're going to calculate the hydrodynamic radius and the size distribution. This is a simple setup we see in all other DLS equipments also. So based on this same parameter only, we are calculating the aggregate size. Another important feature here is that we are understanding the polydispersity also, whether the molecule is monodispersed or polydispersed. So when you have a monodispersed sample, you will get straight simple peaks without any other interferences. But when the sample is highly polydispersed, that means there is a heterogeneous mix. So you get different peaks of different sizes. So that's how you understand whether you have a heterogeneous or a homogeneous sample. As I told you, we are understanding the biologics workflow. So this equipment, where we can use it is important because we are actually understanding all three characterization portfolios, like the aggregation, the thermal melt, and the size. So during the developmental stage or the downstream processing or the formulation, pre-formulation, or in the stability characterization field, or when you are actually comparing batch to batch products, then you can actually understand all these three parameters in a very defined and a very very promising way. Now, as my talk was around multi-parameter stability characterization, so I have few examples here which I can tell you how we have used the equipment in analyzing the molecule of interest. For example, this is one monoclonal antibody we have studied in 3 different buffers. So as I told you, we are understanding it in 3 different parameters, TMs, aggregation, and the size distribution. So you can see in 3 different parameters, in 3 different buffers, the same monoclonal antibody, which is a Herceptin, is behaving differently. So if you see, I have defined all the 3 buffers in 3 different colors, PBS1 in black, the sodium acetate with salt in purple, and the sodium acetate with a pH in blue. And from the TMs, you can make out that the PBS is quite stable because the stability starts, the TM1 is around 65 to 68. So that's how we measure that, okay, fine, my PBS buffer is quite stable. Now, when you look into the aggregation data, you see that around 80, the aggregation starts for the PBS buffer. And for the other buffers, it's still not shown any aggregation. So the idea of understanding the TM and the aggregation lies here, because if TM starts aggregating at 80 and the others are not aggregating, we still can say that my sample can be stable in other buffers also. But now we have to understand whether there are any smaller aggregates or not. So we look into the DLS data. For me, if I look into the DLS data, I can see that even though my PBS is showing a high aggregation ratio, somewhere around 80, the purple sample, which was in sodium acetate with salt, started aggregating somewhere around 62 itself. But the sample which was in sodium acetate at pH 4, it's still not aggregating. So from this, I can actually make out, even though the TMs are quite low, but there is no aggregation till 90, which can be a very good, very stable compound because it's not aggregating. And we have to look into aggregation, because if the molecules start aggregation, then it's not a good molecule to be used into a therapeutic of a therapeutic value. So this is how we actually access the whole parameters. Next slides are actually differentiating individually. So I will just skip these 3 slides. And I will just go into how the data is interpreted. So you can see all 3 data are interpreted by giving their respective TMs and their respective turbidity, which is the aggregation temperature, and the respective hydrodynamic radius at different temperatures, or we can understand the size of each of the aggregate that we are calculating here. Next important thing here is when you are understanding which buffers we have to consider. For example, we have taken 2 different molecules here, a very pure NIST map and a complex sample, which is a complex of antibodies only. And you can see the pure molecule has a very defined peak, which is a hydrodynamic radius of 5.5, and a complex MIS is having a peak which is very broad of 6.9 hydrodynamic radius. And you can see the PDI is also quite disturbed. So when we talk about PDI, a ratio of 0.01 to 0.1, we say it as a monodispersed sample. So that is very homogeneous. And a PDI above 0.2 above is always referred as a polydispersed sample. So that means it's a heterogeneous complex. That is what we have seen, because this is a complex mix of different antibodies. We can also do some stress control studies also. For example, this is a Herceptin molecule, which is in 2 defined buffers, histidine and sodium acetate buffer at different pHs. Initial studies shows that they behave very nicely, but given a stress condition, you can see how the profile changes. So the histidine buffer actually changed, and you can get a very defined, very broad radius. So that means the polydispersity can be seen here. So this kind of stress studies you can do. Then another important parameter here is understanding the self-interaction, which I told you that is also a very important aspect to characterize your molecule, at what concentration it's aggregating. So a KD parameter generally tell you whether it's aggregating or not aggregating, in which concentration and in which conditions. So here also we have defined two different buffer conditions, same Herceptin molecule, and we have studied at different concentration how the self-interaction parameter is analyzed. So when it's aggregating, you will get a negative KD value, and when it's not aggregating, you will get a very positive KD value. That means it's not interacting, and a negative KD value refers that it's interacting with each other, forming large aggregates. So that's how we can define the formulation buffer separately. This is also one important aspect we can understand. Based on TM, we generally understand whether the molecule is in a bound unbound form. So to understand whether the antibody drug conjugate formed is a stable or a potent drug or not. We have already studied this with TMs, but when we look into the size also, we can understand whether it's bound or unbound, which will give you a credibility that yes, my molecule is quite stable, or the drug conjugate that has to be put into other trials is a potent drug target. This is a quite good example where we all know the structure of ATP, how it is, the HSP molecule, how it is. HSP90 is very compact and closed molecule when it binds to a ATP. So we know the hydrodynamic radius will change when it's in an open form. So you can see here in the size aggregation, size studies, when it's in an open form, it has a different radii, but when it's in a closed form, it has a different radii. So that means when it's in a closed form, it's bound and it's a stable molecule, the Ri changes. So that actually reflects the TM and the RH value, what we have got here. So these are all the different parameters that you can study with the same technology. I have a good example here. We have done in collaboration with a very good company called 2Bind, which actually works with vaccines. And they have used their workflow using Prometheus Panda, where they have used each and every parameters to identify their molecule of interest. It will be a quick overview. So first what they have done is they have understood the thermal profile of each of the molecule they have taken into understanding. So you can see they have started from the least stable molecule to a highly stable molecule, and based on these thermal profiles, they have actually taken around 300 plus molecules, and based on these thermal profiles, they actually concluded around 3 molecules of interest, and they want to show how these 3 buffers quite differs with their study of interest. So what they have seen is they have taken 3 buffers, buffer 9, 57, and 70, which are defined in different ratios in the TML. So you can see buffer 70 is least stable, whereas buffer 9 is quite stable. But when you look into the size, you can see that buffer 57 is actually one of the stable molecules, which is giving you a perfect size distribution peak of a size distribution of a homogeneous sample, whereas buffer 70 and buffer 9, they have different particles which are larger, and they have buffer additives also. So that is how they differentiated between these 3 buffers, which they took based on their stability point of view, that is TMs. And then once the TMs are analyzed, they want to access whether these 3 buffers can be used further into the studies or not. But when they analyzed the DLS part of it, they got to know that the buffer 57 was more potent to be used as a good therapeutic buffer rather than the buffer 9 and buffer 70, because they were forming different larger aggregates. They also did some stress control studies where they have considered 3 different temperatures, 25, that is the room temperature, and different conditions like a higher temperature of 40 and 60. And you can see based on the different increase of the temperature, the PDI and the radius changes. So that is the cumulant radius and the polydispersity changes. So this is also one important part to understand, because when the PDI and the radius changes with respect to time, that means it's aggregating, and your formulations can't be used further for any analysis. So this is important when you're giving a stress condition, how your molecule is behaving. As I was telling about the bound and bound nature, they understood their molecule with this also, where bound nature is showing a different, hydrodynamics radius. And when they compared it with a molecule which doesn't interact, you see a very straight line, which is not actually interacting. That is, the RH is not increasing. So this kind of parameters you can easily access when you are actually stabilizing, you're characterizing your molecule in through different parameters. So this whole idea of understanding a particular vaccine of interest or a particular molecule of interest, they took around 2 to 3 weeks, and very small volumes and very small concentrations of samples they have used. And with molecule characterization of almost 300 plus samples, variants, they actually got their working done in almost 2 to 3 weeks, and they got the best product, which they are now using there for other under trial processes. So this was the case study that I wanted to reflect. Other than that, the software is also very unique, very user friendly. Each and every parameter is very easily to understand, and there are booklets, or what we call it as nanopedia, like an encyclopedia, to understand each and every aspect of whatever you are analyzing. For example, if you want to understand what is size analysis, or what is thermal unfolding, or what is DLS, each and everything will be there as a booklet which you can read and access the software in a very user friendly way. This is all about what I have. So I have 2 more slides to just represent what my company is all about. We started our operations in India in 2016, and Shivramiya Nalapeta is heading the team, and the application scientist for the company. And we have, as the product portfolio I showed you, we have one for the binding interaction studies, and one for the stability characterization parameter. Both the products we have here in our World Trade Center office in Bangalore. And till now, we have almost 25 installations in India across academics and in the industry. And based on these different ideas of how to understand the molecule of interest, we have till date almost 55 publications, which are in reputed journals. And if at any point of time anyone want to understand how the system works, or they want to access any molecule of their interest, we are always open for some proof of concept studies, so that we can always help them with understanding their molecule of interest, and also we can do some collaboration work. So this is all I have to say about my talk. You can contact me anytime. My mail ID is also written here. So any questions, I'm happy to take. Thank you.
Yeah. Thank you, Saji, for a wonderful presentation. Can I request you to stop the sharing so that we can display your video? Yeah.
Yes,
thank you.
I'll do that. I've shopped this. Yeah.
Yes. Over to you, Purushottam. Yeah.
Yeah, thank you, Dr. Saji. Really interesting talk and details, especially understanding the protein stability and quick screening of formulations. Is still a lot of area of interest in the new drug discovery and development.
Thank you.
I don't see any questions here yet. I would like to really request participants to really post your questions. Dr. Saji would be happy to clarify some of the technical details or questions.
I think there is one thing. What is the particle size range of this instrument? Yeah. Generally, we have measured from a range of 0.05 nanometer to 1000 nanometer. And that is like 0.05 we have measured for lysozyme. That is we have datas also which we can share it with you. So the lowest range that we have measured with this equipment is 0.05 nanometer.
So, Dr. Saji, while questions getting populated, one question from my side to really understand
Yes.
this Panda instrument, say DLS, combination of DLS and understanding the thermal and thermal folding.
Yeah.
How do you really correlate with the data that comes out from microflow imaging technology? MFI?
Can you please elaborate about that particular technology, because I'm not so aware about that. But DLS is general. This Panta instrument, what we have, we compare it with the normal DLS, what we have, the existing DLS. And if we compare both the technologies together, the data is almost same. The another important aspect here is we don't have to do any buffer, study also, because separately we don't have to understand a buffer component and do the studies, because here what we are doing is we are measuring the TMs, and TMs, whatever data we get from TMs or the aggregation, we correlate that data with the DLS, the same DLS technology what is used in the various other DLS equipments what we have in the existing right now.
Yeah.
So when we are doing that kind of study, we, for example, if we are getting an aggregate somewhere around 60 degree, and the TM is also somewhere around 60, 70. So we need to understand whether there is an aggregate at that point of time or not. But when we see the aggregates, start happening earlier also. So somewhere at 55 itself, the aggregates start. Now from that point, we can understand, okay, my sample has started aggregated somewhere at 55. And what is the size of the aggregate? So we understand the size of the aggregate. For example, if it's a 2 nanometer or 100 nanometer or 1000 nanometer, what kind of aggregate size we have, and whether the aggregates are homogeneous or heterogeneous. For example, if it's a homogeneous, we get a very steep, a very straight peak, or when it's a heterogeneous, we get a very broad peak. So it defines, like, for example, if it's starting from 10 nanometer to 100 nanometer. So we know that there is a heterogeneous peak having different aggregate sizes from 10 nanometer to 100 nanometer. So this data is very much comparable with the existing DLS technique. So what data we get from Panta, we can compare it with the DLS, the existing DLS one.
Yeah. Thanks, Dr. Saji, for clarifying that.
Can we set the temperature of flow cell to study the temperature effect on protein aggregation? What is the range we can set? So the temperature range that we have here is from 15 to 95 with an upgradation to 110 degrees centigrade. So we can understand protein aggregation based on that temperature, what we are giving. I hope I'm correct with what I'm saying, or is there anything that you want to specifically know?
So I think the question is like, can we set the different temperature ranges to really understand the protein aggregation?
Yes, when we are doing a DLS alone, standalone DLS, we can understand the protein at different temperature. For example, we have to understand at 25 degree at room temperature, and then we can understand at different temperatures, for example. So I told you the temperature is defined from 15 to 110. So you can understand the aggregate size from 15 to 110 at any temperature. In standalone, you understand at one particular temperature, like 15, 16, 17, like that you can understand. But when you're doing unfolding, you understand the whole DLS from a range of 15 degree to 110 degree. That's what I showed you in my slide also, how we can understand the TM, the aggregation, and the DLS at a refined temperature range from 15 to 110. I hope I gave the answer.
Thank you, Dr. Saji, for a wonderful talk and a very interactive session. So may I request Purushottam, if there are no further questions, would you like to felicitate her?
Yeah. Thank you. Thank you. Really appreciate for sharing your experience on Panta instruments, and really appreciate for sharing some data, because that's very valuable to understand how to really understand on the protein stability and some of the pre-formulation aspects from this.
Thank you so much, sir. Thank you.
Thank you, Saji, and always been a pleasure to have you in the conference. Thank you, Saji.
Yeah, we are also highly obliged for that. Thank you so much, sir.
Thank you. Over to you, Purushottam.
Yeah, so we have next speaker. Again, very important talk in terms of how do we apply the PAT technology in the crystallization process development. Today we have Dr. Sri Vidya to share her experience and expertise in PAT, and especially crystallization process development. Dr. Sri Vidya Ramakrishnan heads the process engineering at Dr. Reddy's laboratories at Hyderabad. She earned her BTech in chemical engineering from Indian Institute of Technology, Madras, and her PhD from Princeton University, USA. Prior to joining Dr. Reddy's, she worked at Unilever Research, followed by Bristol Myers Squibb in New Jersey. She has also presented her research in international conferences, has several patents and publications to her credit, and is also an ASQ certified Six Sigma Black Belt. Since last year, she has taken up an additional role of Chief Diversity Officer at Dr. Reddy's. She mentors women within the organization as well as external. Is Sashak scholar. Sorry for that. Now, Dr. Reddy's Foundation for Women and Students. So, I'd like to welcome Dr. Sri Vidya to really present her work in the area of crystallization process development and how do we apply the PAT technology. Over to you, Dr. Sri Vidya.
Yeah, hi, good morning, everyone. Am I audible?
Yeah, it's coming clear, but I think maybe a bandwidth issue on the internet.
Dr. Srividya, I think there is a network issue at your end. I can see those yellow. The bandwidth is a little weak, but we can hear you. You can share your screen.
You can hear me, right? Okay, because I think we have a full internet connection, but okay. So it's you're able to hear me and it's clear, right?
Yes, now it is clear. Now I can see the clear this thing, but it's wavy a little bit. I can see the connection there. So we'll do one thing, Dr. Srividya. You can share your screen and you can switch off your video. So maybe we will be able to hear you clear then.
Okay, sure. I'll just yeah. Okay. Yeah. Hi. Good morning, everyone. Again, the slideshow. Yeah. Are you able to see my presentation?
Yes, ma'am. Now it's perfect, and we can hear you clear also. So let's continue this way. No worries. Thank you.
Okay. Sure. Sounds good. All right. So yeah, today I'll be talking about the application of PAT, which is process analytical technologies in crystallization. And in the pharmaceutical industry, as you're aware, crystallization plays a very important role because that's how we design the powder properties of the active pharmaceutical ingredients, the API, and which impacts, bioavailability, the formulation processability, and so on. So understanding your crystallization process is critical to being able to design the powder properties that you wish for in your API. And today I'll touch briefly upon what is PAT and what are the PAT tools that we have for crystallization. One sec, I'll just get my pointer. For crystallization, and I'll follow with 3 case studies. The case studies would be on different kinds of applications. So one is, where we had to address a poor filtration issue. The second one, where we actually wanted to predict solute concentration during a reaction, where offline sampling was not possible. And the third is to optimize polymer transformation. So these are different solid state forms of the API, and we wanted to target a particular solid form, and where PAT helped us to, tailor the process for the, or rather optimize the process. Okay, so what is PAT? It's a system for designing, analyzing, and controlling pharmaceutical manufacturing through timely measurements of the critical quality and performance attributes, right, of the raw materials, in-process materials, and the processes with the goal of ensuring final product quality, right? And there is an, ICH guidance on this as well. But very simply, right, if based on the process understanding and the process control, right, if both are low, we have a high potential of failure. Whereas if both are high, we have a very robust and reproducible process. And both are required because if, say your process understanding is good, you have a good process, but sometimes you detect problems only after they occur. If you have very good process control without good process understanding, you will have a reproducible process, but very narrow operating ranges. So ideally, we want a situation where we are in this green box. And PAT plays a very critical role in helping us develop both process understanding as well as process control. So there are many sensors that many people might have used, and the previous talk was also talking about measurements with light scattering and so on. So there are a lot of measurements on purity, pH, temperature, and so on, which are scalar quantities. But we also have what's multivariate quantities, right? Like spectroscopy, typically, infrared, UV visible, Raman, all these are spectroscopic techniques, which give us a lot of information. And we use chemometrics in order to decode and get the relevant process parameters for us. And I'll touch briefly on chemometrics in one of my examples today. And there are various, tools that we can use in chemometrics, and multi-curve resolution, multivariate regression, and so on. And finally, process control. Often process control is, an outcome by coupling this with, coupling your process understanding with control through automation, feedback control, and so on. So very briefly, with process analytical technologies, we get much faster and more in-depth, I would say, for process understanding, right? With minimal experimentation, we can run one experiment, which gives us a lot of information about what's happening in our process. And like I say, in process control through, say, feedback control, even predictive modeling, all of that. And today, the data science is really, becoming a hot topic. So a lot of this data can also be used into machine learning algorithms. And we also want in situ analytics for process monitoring. So, sometimes we are not able to access information about a particular reaction species, which can be done only through in situ sampling. Yeah. And in the manufacturing setup, people also use PAT for real-time release. Today, as continuous manufacturing becomes a very hot topic, real-time release, like you actually have your PAT in line with your continuous manufacturing line, and you can have real-time release of your product based on the data. So I'll get into PAT for crystallization process development. Two of the tools that we use a lot are PVM. This is a particle vision microscope, where you insert a probe in your reactor, and it's basically a microscope, right? It's giving you pictures of what's happening. How are the crystals looking inside the reactor? And there's a lot of, understanding. To me, a picture is always worth 1,000 words, right? So pictures can really tell you what's happening in your process. The second is FBRM, that's focused beam reflectance measurement. And what this measures is a Cordillon distribution. So let's say if this is a particle, there's a laser beam that goes across, and this distance that is cutting, going through, where there's an obscuration because of the particle, that is basically the chord length. Yeah, and so you have this, high velocity scanning beam that's detecting a number of particles, and what it gives you is a distribution. And from that, we can get counts. For example, the red line here is number of counts below 10 microns. So that it's a number of particles it's counting within a certain period of time as it scans. A number of particles between 10 to 50, a number of particles between 50 to 150. So we can get also a count of it. And how the count evolves over a period of time. So in this case, for example, there was no crystallization here, and suddenly there was nucleation. And so all the counts increased, and then they're gradually growing with time. We also use other spectroscopic techniques like Raman, and Raman can give us a lot of information on the solid state form of the API. So for example, here you have one particular form, form 2, and this is another form, form 1. And this is the solute concentration. So, the solute concentration is dropping as this blue one, the form 2 crystallizes, and then form 2 starts converting to form 1. So you see the form 2 peak going down, and the form 1 peak starts increasing at this point. So you're not only able to monitor your concentration in solution, but also what's happening to the various solid forms that are crystallizing out. And we often use this to study the kinetics of a form transformation, and this will be one of the examples I talk about today. You also have FTIR and UV-Vis, where, based on the various functional groups that are present, and their, whether it's stretching or bending the various frequencies, we will get a very characteristic spectroscopic data of, the species that are in solution. So these will typically measure what is in solution. Raman has the advantage that it will also measure what is crystallizing out. So with this, I'll move to the first case study. And in this case study, we have an API, which is a salt, which is dissolved in toluene. And there's a small amount of methanol added because we want to get rid of a particular styrene impurity, and a bit of methanol helps to wash it out. And then there's a seeding and there's a crystallization that happens here, right? So it's dissolved at 82 degrees. And, the seeds were added, and then it was cooled to 20 to 25 degrees. And after that, it proceeded for filtration. And during filtration, there is a washing that's given to the wet cake, followed by drying under vacuum at 35 degrees. So this is a very typical crystallization process. The big challenge here was that the filtration was extremely slow. Yeah, and I'm sorry, I think there's an error in the celluloid addition temperature. It should be about 70 to 65, I think around 70 to 75 degrees, but we'll come back to it. So it's a very slow filtration, right? It took forever. And then after the first crystallization, there were 4 recrystallization steps just to wash off the styrene impurity. So just crystallizing it over and over again. And you can imagine that in each crystallization, we would be losing a lot of yield. And also you have a mixture of solvents like methanol and toluene, which makes separation and solvent recovery a little bit more challenging. Ideally, we would prefer to have a single solvent, which we can readily recover. So we studied this process with Pat. And our first thing was to, what is the optimum seeding temperature? And typically slow filtration is an outcome of, very fine crystals. So can we try to grow the crystals during the crystallization? So this was the process as it was given to us. Yeah, like I said, the seed was added at 75 degrees. And what you're seeing here is the green one is the solute concentration. Yeah, so once it's, you've dissolved your API in toluene, and this is a solute concentration that you're monitoring here, and the blue is the temperature, right, which is being cooled in steps. So at 75 degrees, the seed is added. Nothing really happens. So there's no nucleation, no further precipitation, happening after seed addition, and the seeds have probably dissolved. And as you cool in steps to 74, 72, and to 68, you see the solute concentration is flat. And then suddenly, and what happens? There is a point in the temperature where there is a sharp drop in the solute concentration, a very, very sharp drop. And we have basically come from a concentration in solution, which was, let's say here about, we don't have the actual absolute numbers here, but if you look at a signal from about 80% to say 30%. So there's a very significant drop. And this is just qualitative data with the React IR. Yeah. Very sharp drop in the solute concentration. And further than that, there isn't much drop as you cool it down. So there is really something happening here between the temperature of, say, 68 degrees to 55 degrees. To further understand this, we studied the process with both FBRM and with React-IR, and you can place both probes in the crystallizer at the same time. So there's a lot of information you get. Here you have dissolution. So here you have particles. They've dissolved. So you don't see any particles anymore. It's a clear solution. And then what happens is that there is, a seed addition at 70 degrees in this case. And when the seeds were added at 70 degrees, we do see nucleation happening. Yeah, we do see nucleation happening. And there is and if you look at the solute concentration, there is a slight drop, right? It's gradual. It's not a very sharp drop, but there is some decrease in the solute concentration as the material crystallizes out. And what's interesting is if you focus on this region, there's again a fairly sharp drop as you continue to cool it. So this is a slow cooling experiment. It's a ramp cooling. So from 70 degrees, we are cooling it to 30 degrees at a rate of 0.1 degrees per minute. And then by this temperature, we are basically at 60 degrees. So if you see between the 70 degrees to 60 degrees is where this drop has happened. Yeah, going on between 70 to 60 degrees. And that's the insight that we could get from monitoring both the FBRM as well as the solute concentration. The FBRM doesn't really tell me that there's a sharp drop in concentration, though it tells me crystallization is happening. It also tells me that, my fines are decreasing and my crystals are growing, right? And you can see here there's further growth in the larger particles, which are 150 to 300 microns. Yeah, but however, it doesn't tell me, FBRM cannot give me information about this sharp drop in the solute concentration, which I could get only through React-IR. So this is really where I want to control my process, because we do not want, when you want to grow crystals, you don't want to suddenly, increase the degree of supersaturation. You want to have a understanding from the last two experiments. Is we ran the third experiment where we said, yeah, we'll seed it around 70, 72 degrees.
Hello, Dr. Srividya.
Seeded it around 72 degrees. We do see as the crystals in a particle. And here what we're doing is, yeah. Hello?
I think your voice is coming and then dropping.
Hello?
Hello.
Yeah, Dr. Srividya, now we can hear you. I mean, just for a moment in between, your voice was feeble again. Now we can hear you. Can you continue? Yeah. No, for some reason, we are still not finding the audio.
Connection. Yeah.
I mean, Dr. Srividya, for most part, actually, your voice was very clear. In last, I would say, last minute or so, it is just we can't hear you.
Hello?
Yes, Dr. Srividya, we can hear you.
Yeah, can you hear me? Is my screen visible or do I need to share it again?
I think, Dr. Srividya, you'll have to share it again. I think the sharing has stopped, so you'll have to share it again.
Okay, 1 second. Yeah, is my screen visible now?
Yes, we can see your screen and your voice is also audible. Please continue.
And are we on the slide?
No, I think your voice is quite feeble, Dr. Srividya.
seeing if that is any better. Yeah. Yeah, is this any better?
Yeah,
Yes.
it's much better now.
This is
It's
much better.
Much better.
Definitely.
Okay, right. Thank you. I'm sorry. There's also a lot of construction work happening here without any, noise adjacent to my complex. So there might be some background noise from this location, but I hope you can hear me better now.
No, your voice is pretty clear now. So please proceed, Dr. Sriva.
Okay,
Thank you.
Sure. All right. So moving further, yeah. So you're on the slide that says optimization of cooling crystallization parameters, right?
That's correct.
Okay, right. So what I was trying to say was, by understanding where the solute concentration drops, we were able to basically design a step cooling where we could, have a very, hold the step right here between 65 to 70 degrees, 65 to 60 degrees, where there's a drop in the solute concentration. Yeah. So, and then we could actually grow the crystals pretty big. I think my slide is not changing. Yeah. And if you see, this is what we had from the process that gave us very poor filtration. And this is the process that we, optimized, which could significantly improve the filtration rate. So from 8 liters per meter squared per minute, that was the filtration flux we had originally. We could actually, improve the filtration flux 10-folds by simply growing the crystals and identifying what is the right, position for seeding, as well as, maintenance, so that we de-supersaturate very slowly. So this is the first case study. The second case study is on quantitative prediction of solute during reactive crystallization and to improve the yield. So in this case, we have our two key starting materials in this reaction, KSM1 and KSM2, which are dissolved in dimethyl estamide in the presence of a base. And then further, once a product is formed, the crystallization happens with the addition of water as an antisolvent. And here there was no analytical method to predict, how much of the product is formed. Are we forming as much product as we need? And, how much are we crystallizing out? And what is the yield that's there? And this is where I talked about chemometrics, where we use, partial least squares to predict the concentration in the solute. So if you look at this data, here's KSM1, that's a blue one, and KSM2 is blue and KSM1 is pink. And both are reacting, and the reaction is, quite rapid initially, but it still continues for a period of about 6 hours. And the green curve is the product that is getting formed, which we can monitor with React IR. Yeah, so we see the product being formed, and at some point the product starts crystallizing out, and there is a drop in the concentration, which goes on, gradually until the point where we add water, and there's a sharper drop in the concentration. Now, this is qualitative data, right? So with each water addition, there's a further drop in concentration. And then there's a cooling that's happening here between 15 to 5 degrees, but you don't see much impact on the cooling. Yeah, mainly the concentration is dropping as we add water. So none of this is quantitative. We don't have any numbers of what's the concentration in solution, what's the final concentration, and so on. And, this is really what we wanted to understand during the process. So here we used partial least squares regression, where, we have a data matrix and a calibration matrix. And basically, there's so much information. What chemometrics does here, right? There's so much information in the spectral data, which then gets, minimized into these principal components. So there's a scores and loading matrix, and I don't want to get into that because anyone can read further on chemometrics. But very simply, we process the data, and then we have a model that is built, after pre-processing this data, and a cross-validation. So we build a calibration curve. So let's say there are 10 points in my calibration curve, right? Let's say 8 or 9 points are used to build the model, and then 1 or 2 points are used to cross-validate. And so then what happens is we now have a model which tells us based on the spectral data what our concentration is. So then when I get a new data point, the chemometrics can predict for me exactly what is the concentration. So this is the idea of the quantitative modeling using partial least squares regression. And here, in this case, we have multiple components in the model. We have the product, we have the KSM1 and KSM2, which we started with. We also have DMAC and water. This is dimethyl estamide and water, and the ratio is also changing during the process. So we start with, zero water, no water initially, but then we keep adding water in different lots. So the dimethyl acetamide to water ratio is also changing. So to factor in all these, components and the solvent mixture composition in these concentration ranges, basically based on where we expect the concentrations to be based on a reaction, we created a data set of 60 samples which were used to build the PLS calibration model. And so this is the data before as it is, and then we pre-processed using a second derivative to cut out the noise and all. And with these 60 samples, we could build the PLS calibration model. And once we have the PLS calibration model, then we could actually quantitate, right? What is the concentration? So this is the root mean square error for cross validation, which is about 26 mg per ml, which is about 7% error, which is not too bad, right? We just want to be, we were pretty close to where we want to be. And what the model predicted, is that the concentration when we have formed all the product is 400 mg per ml. And based on how many moles of the reactants we took, we know that the maximum theoretical product concentration is 430 mg per ml. So our model is pretty good. We are predicting in the right range. And during this entire, crystallization of the product, not in the crystallization, the formation of the product and its crystallization, we are able to quantitate the concentration at any point in time during this reaction and crystallization. Right, so we know exact, and you can see there's a sharp drop to 126 mg per ml of the first water is added, then to 68 mg per ml when the second lot of water is added, and the final concentration is 45 mg per ml. And with this, we could also optimize how much water we want to add to improve the yield. And with this kind of quantification, we can also build kinetic models for the reaction and so on, and even for the crystallization. So I would say this is a very useful technique to progress from qualitative to quantitative models. And how am I doing on time? Should I take the third case study, or should I stop for questions?
It's okay, ma'am. You can just complete this and then we'll take up the questions.
Okay, this is my final case study. I'll just be quick about it. So this is what I talked about, understanding the kinetics of polymorphic transformation. So the last two case studies, we talked about reactor and FBRM, and here I'll be talking about Raman. So we have an MIBK solvate of this API. Right, that means MIBK is part of the crystal lattice. So this is the crystal form that we start with. And we want to convert it to an anhydrous form, which is form C. And what we do is we just make a slurry in isopropyl alcohol at 30 degrees. And over a period of time, this solvate converts to this anhydrous form, which is the desired targeted form that we want to have as the final API. So if you look here, the blue depicts the MIBK solvate. This is with Raman, right? So from Raman spectra, we are able to pick out characteristic peaks of the MIBK solvate and characteristic peaks of the anhydrous form. And this is basically now looking at the solid. So while reactor, again, I want to emphasize, while reactor was looking at the concentration solution, now Raman here is looking at the solid, right? What is the solid form of the solid that's coming out? So this is, if you see at 25 degrees, if I'm just maintaining a slurry in isopropyl alcohol, and this x-axis is over a period of time, right? For a long period, for nearly 5 hours, nothing really happens. And after that, this form slowly starts converting. So you see MIBK solvate drop. And at the same time, you see the increase in the form C. So MIBK solvate is converting to form C. Now we wanted to kind of make this happen a little faster. So we used much finer particles. So we micronized this API, this MIBK solvate, and then did the same slurry at the same temperature. And what we find is immediately the form starts converting. So immediately MIBK solvate is going down and the form C is going up. So by decreasing the particle size, we could accelerate the kinetics of form conversion. We also studied the effect of seeding. This is with the original particle size, non-micronized. We added 3% seeds, and we could reduce the time again for the initiation of the conversion. So within 2.5 hours, we could convert it from MIBK solvate to form C. And this is very useful to us because we can now monitor through this. How much time do I need to convert it to form C? And what can I do in the process, right? What is the impact of, let's say, a finer particle size? What's the impact of seeding? Here we studied the impact of temperature. So at lower temperature, it took us much longer to convert it. So and at higher temperature, we could and with seeding, we could convert it fairly, fast. So we could basically optimize our process by studying these kinetics of form transformation using Raman. Yeah, otherwise, if you just imagine to get this much data of exactly how the form is transforming, you have to keep pulling samples, keep checking the XRD, which is very also, very labor intensive, to be frank, right? So much effort goes into just keeping on pulling samples and running the XRD. And all of this we get by just inserting a probe, and the information is readily available to us. So with this, like I said, we were able to optimize our process. And I want to thank everyone. So even at the beginning of my talk, I missed out, but just to go back to the very beginning, I would first like to acknowledge, my co-authors, Kamla Jyotsna, who put a lot of effort into, preparing this entire slide deck, and Ninad also, who heads the PAT team at Dr. Reddy's. So I'd like to thank both of them for, all their help in putting together this talk and, coming up with all these case studies. And they just do brilliant work, on so many processes with this. And and all my other colleagues who worked on these projects, Chiranjeevi, Sesha Reddy, Narendra, and Monica, who did the chemometrics model. So with this, thank you very much. And I look forward to any questions.
Yeah, thank you, Dr. Srividya. Very interesting talk, and sharing a deeper insight to the crystallization process and how one should look at different parameters to optimize and get the right outcome where we are looking forward. I would like to request all the participants to share their questions. Not able to see yet, but maybe one just insight, Dr. Srividya, especially on the last case study, what you shared on converting the polymorphic form to say a solvate form to anhydrous. Especially when you look at the real-time PAT analytics, what's the sensitivity of detection? You'd like to really throw your experience in terms of having the solvate in the amorphous or any crystalline form in the amorphous. Of course, the isolated analysis can throw you in terms of lowest percentage of detection. But in a PAT online process, what's your experience in those sensitivities of percentage?
Yeah. I mean, online analysis is never going to be as sensitive as the offline analysis, but I think it's great for getting the information, through the process. So here, I don't remember in this exact case, it could be around maybe say 5% or so, right? That's typical, but we cannot get the kind of sensitivities that we would get with XRD, with a slow scan in the particular wavelength and so on. But with chemometrics though, I think the sensitivity is increasing. So what I showed you now in terms of the kinetics for polymorph was qualitative. But again, like the previous example, we can apply chemometrics to Raman data as well to build more quantitative information.
No, absolutely, yes. Chemometrics, once you build that calibration and the further details coming out, yes.
So I think I don't see any more questions.
There is one question in the Q&A tab. If you'd like to take up, Purushottam.
Not able to see. I don't know, something maybe.
Okay. So under the Q&A tab, the question is from Mandar Manulikar. The PAT tools are typically useful on SAT processes. Is that a correct understanding? And secondly, can any such in situ tools be useful during the process development?
No, sorry. The first question is what PAT tools are typically
useful on set processes. So is that a correct understanding?
No. PAT tools, you actually want to use them before you set your process, right? So it depends on what you're using PAT tools for. So like I said in the beginning, right, if you want to use PAT tools, and it's related to your second question as well, if you want to use PAT tools for process understanding during process development, You do not need a set process. You can simply insert a PAT tool at the very beginning, in your very first experiment, to understand what is happening in your system. It will give you a lot of insight and understanding. It will minimize experimentation time. It will minimize the number of experiments you have to carry out, because you get so much insight. I mean, like the crystallization one, I showed you 3 experiments, right? 3 experiments where we could, identify where it's, where the concentration is suddenly dropping, which temperature range do I need to focus on, all that in just 3 experiments, and I've optimized my process. So you can seriously use PAT tools at the very beginning. You do not need a set process for it. You use it for process understanding during development. And now if you're saying, I want to go to manufacturing and I want to use PAT tools for control or for real-time release, then that is a different application, which I have not discussed it today. And that also needs much more, your models, maintenance of models, and with, all the regulatory and all of this. But that's a different process where, application we're using it for set processes. But during R&D in development, you would very much use it on processes for they're very much in development and for understanding. Yeah.
Thank you, Dr. Srividya. And I think I agree completely. If you use this PAT earlier, even before the SAT processes, that would really give an advantage of having it actually at first place. Thank you. Any further questions from you, Purushottam?
No, I don't see any questions, but maybe one more question from my side, Dr. Srividya. If you'd like to throw a light on in terms of where the industry is moving in terms of real-time release of API and drug product, especially using this PAT technology, chemometrics, and other aspects of it. Maybe you'd like to share some experience to the audience.
Yeah, sure. I think the industry is getting more into real-time release, especially on the drug product side.
Yeah.
And particularly for continuous manufacturing, because you have continuous manufacturing lines where you do want assurance on the quality, right, as your product comes out of the continuous manufacturing line. And they also have a divert to waste strategy, where if your product is not meeting the desired quality, there would be an automatic feedback control where the product will get diverted to waste. So that way it's basically real-time release testing that says, hey, if my quality attributes comply, then I'm going to collect it in one bin. If they don't comply, I'm going to collect it in a different bin. So I think that way it's picking up a lot. And we are looking at various PAT, even on API, I'd say, in manufacturing for some of this, like the React IR can be used in manufacturing. There's also Waters Patrol UPLC, which can automatically pick up samples every few minutes based on your method and give you chromatograph of your purity profile. So I'll say the industry is definitely picking up in this aspect. Entire real-time release testing may not be possible, but partially yes, at least for some of the key quality attributes.
Yeah, thanks, Dr. Srividya. Yeah, because the industry is really looking at, in fact, right from starting material, your API first manufacturing step, to getting the product released to the market. How it can go into a real time?
Absolutely.
And that's the area picking up in the industry.
Yeah.
Really, once again, thank you for your interesting talk and sharing valuable information.
Thank you. Yeah, thank you very much. Really appreciate it. And happy to be part of this forum. And thanks so much for inviting me.
Yeah. Maybe.
Thank you, Dr. Srividya, and for a wonderful talk. And we would like to extend invitation to you in advance. Next year, hopefully, we'll be doing in-person meeting. We'll be more than happy to have you in person delivering a talk. Thank you, Dr. Srividya.
Definitely. Thank you very much. Yeah. Bye-bye.
Yeah, over to you, Purushottam.
So,
we'll move on to the last.
So today's our last session, especially on bridging the technology advances in regulated bioanalytics. I'd like to invite Dr. Kuldeep to share his experience and his knowledge to all of us, especially on the regulated bioanalysis. A very quick introduction about Dr. Kuldeep Sharma. He has over 19 years of experience in area of bioanalytical research analytical research and development, drug metabolism, pharmacokinetics, product marketing, market research and pharma regulatory compliance. Very diverse experience, I would say, to share with us. And his journey covers a true blend of technical and commercial industry insight over past 12 years in analytical research. Uh, in his current role as a sales development manager at Agilent Technologies, his core responsibilities include business planning, marketing research, sales forecast, competition analysis, sales and enablement, strategy planning, and execution. During his industry experience, he has accomplished leading roles in domains covering conduct of analytical research, preclinical, pharmacokinetic and toxicokinetic studies, in vitro analysis, tissue distribution, and in vivo metabolic profiling. With over 21 plus research articles published in the international journals, like Journal of Bioanalysis, Anali di Chimica, Journal of Brazilian and Chemical Society, and Journal of Chromatography B. I would like to invite Dr. Kuldeep to share his experience in technological advances, especially from a regulated bioanalysis perspective.
Thank
Over
you.
to you,
Thank
Dr.
you. Thank you, Dr. Purushottam. Let me share my screen. Once again, let me put it in presentation mode.
Yeah.
So I believe my slides are in presentation mode.
Yes, it's in presentation mode and we can see those.
Thank you. Thank you for confirming. So it's last session of today's web series, and I'm really delighted to be part of APA 2021 virtual web series, sharing some insights about the recent technology development. So when I started thinking about my talk, basic objective which I was having in my mind is to bridge in the technology advances with the requirements which we have in bioanalysis. So I'll be covering upon some instrument features and how exactly they will be beneficial for the scientist in taking up their research work. So first, talking about hardware. So there definitely Agilent has some unique hardware features in their mass spectrometers as well as HPLCs, which give a true optimum performance for any bioanalytical application, like wide mass bandwidth, which we have till around 3000 mass by charge unit. Then with UPLCs, we have a fast injection to injection cycle, as well as good reproducibility, which actually gives the unique combination a true productive outcome. And as I said, consistent, predictable performance, which is definitely a recipe for Agilent being into this arena for a long time. Now coming on to software part, because that is also an integral part of the complete workflow. The data which we get from the hardware has to be in a format which is easily understandable to the user, as well as they are able to actually take out some messages very quick to decide onto their workflows. So Agilent Mass Hunter features I'll be talking upon, which actually which gives the users a very simple interface to understand the results outcome, and then also with respect to compliance, because if we don't have a compliance in between these two components, hardware and software, then the overall objective of regulated workflow is lost. So I'll be talking on to these three parts. I have some case studies also to show you that how exactly the hardware features are contributing in the overall results. So Agilent has a wide portfolio. When I say wide portfolio, probably it covers the complete workflow for analytical lab. Starting from the sample preparation, we have some cleanup tools which can help reducing the matrix component and excipient effect onto your analysis. Then we have columns. Poroshell columns are well known in industry for their rugged performance and long life. Then with respect to HPLCs and UPLCs, we have 1260 and 1290. In 1260 also, we have a very new prime edition, which can go up to a higher pressure limits also. Coming on to MS, I'll be talking on 6470 and 6490 basically to give you some insights that what features exactly make these instruments very unique in industry. So coming on to the UHPLC first, what makes Agilent UPLCs stand out in the market? First is the multi-sampler. So in the multi-sampler, actually, we can go up to 6 well plates configuration. And when I talk of 6 well plates, and we are talking about, let's say 384 well plates to be put in to each plate, we are talking about around 6,144 samples to go in one go. So it definitely can cater to the kind of fast speed and fast analysis which we expect from the mass spectrometers of today. So such kind of unique combinations from auto sampler, as well as it has a flow through needle design which gives less carryover and zero injection delay, probably because of the dual needle design which we have put into this. I'll talk on that part very little in the next slide. Then comes the column compartment. As you can see very clearly, we can put up to 8 different columns. So this is very good for method development part, because many a times scientists struggle to screen in different stationary phases. So no need to actually sit with instrument and wait until the one analysis is over. They can just put in multiple columns, different stationary phases into this setup, and leave the instrument overnight running to screen all the possible combinations or chemistries which they want to screen in for. The third one over here is the Quick Connect. So Quick Connect is a unique product which Agilent has, and it basically gives a tool-free connection to the analytical columns and has a zero dead volume specification, which is very good for any chromatographer who daily struggles with spanners to change columns from one application to another. Quick change walls and quick connect heat exchangers are some other features which give a optimum performance to the UPLCs. Now, quick connect heat exchangers basically are the heating components which can preheat the mobile phase before it enters into the analytical column. As I said in my previous slide, the dual needle concept. So in UPLCs, we can go with the dual needle concept. So as you can clearly see, the gray versus the yellow graph over here shows the injection to injection delay impact. Usually what happens is that from one analysis to another, there is a gap of some seconds for the injector to wash off and then go to the next vial. But that can be overcome by using a dual needle concept. Also, Agilent focuses on to environment impact also. So we actually have a ACT label by My Green Lab, which shows that not only in our products, we also are very committed in our manufacturing to achieve a zero pollution target with respect to our instruments. The unique feature of Agilent technology is a modular design in which for future upgrades, you need not to completely change the complete instrument. Only with the changing modules, you can actually achieve any upgraded performance level also. Coming on to 6470 triple quad instruments, MS. So this is the triple quad MS1, which I have picked up for today's discussion. So as you can see over here, Agilent Jetstream is one unique feature which improves the focus of the spray onto the orifice. Then dielectric injector channel, which actually improves the elimination of any neutrals and other charges and focuses the ion beam also. Octopole ion guide has 8 octopole channels to focus the ion beam. Then vac shield technology is something unique. I'll talk on this later in one slide, which gives a unique advantage of cleaning the instrument, because many a times when we have the instrument dirty, we actually have to break the vacuum to clean the optics. So such kind of breaking vacuum actually leads to a very long delay in bringing the vacuum back to the normal state of operation. So that can be avoided by vac shield technology. Within, I think 30 minutes, we can upbring the instrument to the desired vacuum levels for a quick analysis. Then tapered curved collision cell. This is also very unique because the lab space, it has two advantages basically why it is curved. So first thing is about the bench space. So bench space definitely reduces when we talk of a curve into the ion optics. It takes lesser space on the bench, as well as it also improves the noise level, because the noise goes down drastically as there is a bend in the ion transition. So that kind of unique features give Agilent a true optimum performance, and that was the probable reason that we also got a scientist award also when we introduced this technology. Coming on to the detectors, we have high energy energy diodes as well as detector horn chambers to give you a very high efficiency of the signals and the ions which reach to the detector, both in positive as well as the negative modes. Usually we see a difference while transiting from positive to negative mode in terms of the response. 6495, most of the features are same in terms of the hardware, but I would like to draw your attention onto two principal differences. One is the hexabore ion injector. So we all know, to improve the response of any mass detector, usually what vendors or engineers do is that they will open up the orifice so that more number of ions actually enter into the ion optics. While doing so, what happens is that we also get the neutrals and unwanted ions going into the mass rail, which creates the higher noise. So that can be reduced to a very significant level to be accurate 10 times, which we have also done through experiment to see that almost 10 times improvement in the sensitivity can be achieved if we are using a hexabore ion injector design. And now coming on to some of videos which will show you how exactly it happens. So as you can see over here in this video right now, we have the jet stream off. So as soon as the spray comes out of the needle, actually, you can see a complete expansion of the spray into the area. But when we switch on the Agilent jet stream, which is nothing but a heated nitrogen seed gas, what happens is that that complete spray, which was expanding as it was coming out of the needle, actually focuses into a narrow zone. So it focuses the spray into one single direction, and that way actually the ions which are coming through that spray actually reach more efficiently or in more population to the orifice where exactly they should be reaching, instead of the conventional design. We have 4 ion source options to choose from: electrospray ionization, which is very common in any LC-MS MS application. We have APCI also as a dedicated source, which can look up to semi-polar and non-polar analysis in a very efficient way. As I said in the previous slide, Agilent jet stream ion source is something which improves the sensitivity to around 10-fold. And we also have a multi-mode ionization for research work, which can give efficiency of both ESI as well as APCI analysis into a single channel. Now talking about the hexabore technology. So as I said, to improve the response, many times what engineers do is that they will open up the orifice to higher size so that more number of ions actually enter into the ion optics. But in that case, what happens is that the unwanted ions also start entering into the ion optics, and thus the noise increases. So what we have done into the hexabore technology is that we have instead of having a single hole, we have actually 6 holes created onto the ion optics, which actually gives a very focused transition of only the selected ions into that ion optics. So not all the ions will be able to travel, only the ions which have a specific ionization for that particular voltage will be able to pass through that channel, and thus way we have a clear segment of ions, or maybe a filtration of ions happening at this stage 1 only. What you see later is ion funnel. So we have 2 ion funnels capped, and at a certain off axis, if you see over here, the ion funnel 1 is slightly off axis from ion funnel 2. So it also filters the ion beam to a second level so that we have better sensitivity or selectivity into our analysis. The last part of the hardware feature which I would like to share with you all is Vac Shield. So as I said, Vac Shield basically is a tool which gives the users a definite advantage in terms of cleaning the ion optics. As the system gets dirty with use, they can actually use this feature to clean the system very quickly. So instead of taking around 6 to 6.5 hours, which is the usual cycle for a cleanup, the complete cleanup can be done within a half an hour or so. Coming on to the case studies, so first case study which I have taken up is donepezil. Donepezil is a Alzheimer's disease drug which is very much in use, and what we have tried to see into this particular molecule is the reproducibility and the dynamic range. So as you can see over here in this chromatogram, this slide, I have tried to show you the calibration curve, which is in the range of 3-fold dynamic linearity, and the regression coefficient was 0.993 for this particular experiment. Coming on to the key component or the key benefit of this hardware. Now, if you see the slide, you can very clearly see that I am not talking about reproducibility with respect to say ratio. I am also talking about the reproducibility with respect to area, a specific area of the drug or the analyte. So very clearly we can see that not only the area ratio, but the area of the drug at LLOQ level is also very, very highly reproducible, which gives a very good percentage CV for multiple injection analysis or long batches. The second application which I have taken up for today's discussion is melatonin. Now, melatonin is an endogenous hormone. So when we talk of endogenous hormones or analytes which actually have an endogenous presence, what exactly troubles us is that how exactly we should define a quantitation strategy. So in this case, we have used a deuterated standard of melatonin for actually getting a sense of the signal, what exactly will be the dynamic linearity for melatonin, and using that signal range, we have tried to correlate it to the endogenous level. So as you can see over here, clearly there was no plasma blank interference for the D4 standard. And then we had also kept a quantifier and a qualifier. So it was a two-way strategy to just ensure that if we are not missing onto any components due to endogenous interferences into the assay. Here you can see the dynamic linearity range. So we are talking about a 5-fold dynamic linearity range, because when we talk of endogenous levels, the basic trouble is with the variations which we see at the basal level. But also as we try to go for a treatment, actually, there is a huge shoot up into the actual levels as we try to see a therapeutic effect due to those increased level of that particular component. So a 5-fold dynamic linearity range was established for melatonin D4. And then we tried to correlate the endogenous levels also into some of the plasma samples. So herein you can very clearly see that first we tried to do a spiking of the plasma with the standard, and then created some experiment to correlate back the endogenous levels of that particular compound. Coming on to the compliance, so this is a white paper. We actually have released a white paper some years back to tell what features of Mass Hunter gives you compliance with respect to 21 CFR Part 11 environment. What are the 3 basic requirements? First is the security of the electronic records as per the 21 CFR. The second requirement as per 21 CFR is attribution of the work. And third one is electronic signatures to track who did what into the complete workflow. Mass Hunter has all these 3 features enabled into the workflow to give you a complete traceability of the 21 CFR Part 11 requirements. What exactly it does is that we have unique user IDs and passwords for every user, as well as we have the role privileges also. So there are basically 3 role privileges: manager, chemist, and operator. And under these 3 roles, we can assign different users from our lab. Audit trails, as I said, we have audit trails for data file, we have audit trails for method, work list, as well as the result tables also. Another feature of Mass Hunter is, hello? Yeah, so another feature of Mass Hunter is a checksum and versioning capability, which actually avoids any possibility of overwriting of any file into the Mass Hunter acquisition. The last feature which I would like to talk about is tracing the data, because many times when we have such kind of huge data sitting into our database, we also want to have a feature that how exactly I can search data files from that much complex database. So some unique features of searching for any specific file or any specific information is also enabled into Mass Hunter. Mass Hunter comes with the EC view option. So when I talk of EC view option, different analysts or chromatographers have different preferences. Like for example, if I am doing a method development, my preference will be to overlay the previous chromatograms and see that what exactly is the difference in the new acquisition. But when I am reviewing the data for a study, my preference will be to see the peak, the concentration, as well as the curve in the one single window. So such kind of unique permutation combinations are possible with some features already captured into the review. So to conclude my talk, we spoke about 1290 unique features like multi-sampler, which can give you high capacity of samples, multi-column thermostat. We also talked about quick connect, which gives us zero delay volume, and dual needle design, which can basically avoid any injection to injection delay. In MS, we have Agilent jet stream, back shield, and hexabore capillary design, which actually differentiates the Agilent hardware from the competition. And Mass Hunter, as I showed you, has some features to enable the 21 CFR Part 11 compliance. That brings me to end of my talk, and thank you. Let me stop sharing and go to the questions.
Thanks, Dr. Kuldeep, really sharing us the recent advances at Agilent. Maybe we'd like to invite a few questions. In the interest of time, we'll be able to take one or two questions, and maybe then later on you can post your questions for Dr. Kuldeep.
No questions means either it is a morning effect or correct.
I think we should take it in a positive note that they are so overwhelmed with the offerings from the Agilent that I think it will take some time to digest that and then come back with some questions.
I am delighted.
So maybe maybe Dr. Kuldeep, on that HPLC part of it, any limitations or challenges you would like to share in terms of the mobile phase heating element, any specific precautions or any aspect we should be aware of?
Yes. So as you would have seen in one of my slide, I had showed you the preheaters also, because many times we worry about the column oven temperature, but we miss on to the part that yes, the mobile phase which is entering into that column actually comes from a room temperature. And usually we have air conditioned environment in labs, which in fact reduces the overall temperature of the solvent to a lower degree. Considering that particular attribute only, what we have done is that we have preheaters also as an option available, which can be installed prior to the column inlet, so that the mobile phase which is entering into the column for separation actually is already preheated to that level, so that we don't have such kind of a temperature difference effect coming during the separation. Because during development, we actually have done so much work to understand the temperature impact onto separation. But eventually, when we are running our experiments, the difference of lab environment or the temperature difference in lab actually can create some unnecessary disturbance into the separation due to this temperature difference of two components, column oven temperature compartment and the mobile phase which is entering.
Yeah, thanks, Dr. Kuldeep. It's very important because the reproducibility sometimes comes into a challenge when you have very closely rotating.
Absolutely, because in short runs, actually, we have to analyze everything within a very short time frame. And if such kind of difference is there, it eventually impacts to a greater extent in separation. I do agree.
Kuldeep, this is Amarnath. I have a question which is off track from your talk. The question is, I think you had a good talk on the new technology, but still, we could see in the India in terms of the discovery lab or in the bioanalytical arena, is still Agilent is far behind to capture the, such kind of the lab. Do you feel that what could be the basic reason is, even if you look at the sites and now the waters and the third, I would say the thermo, but still Agilent is far behind to capture these markets. So what do you think that, since you are in the sales, I would like to understand from your perspective that what could be the potential reason why Agilent is still far behind?
This is a question which is very close to my work plan also. And I will agree that yes, in terms of market share, if we talk in regulated bioanalysis, SciEx definitely has the biggest share. And then we have Waters, and now Agilent is also making some inroads into bioanalysis. There are two reasons for this, sir. The first is when industry started booming, that time it was only one single vendor. And that particularly gave a very good opportunity for that single vendor to take this huge market share. If we see globally, that is not the case. Okay, so this is only with respect to India, the channel, the services, as well as a sudden boom into the market gave such kind of a market share advantage. Now, with that particular attribute, what happens is the users get acquainted with that workflow. So if I am comfortable in using Android, I may probably take a step back when you talk about means high-fi features of Apple or some other platform. So such kind of transition from one workflow to another workflow requires some very unique confidence in the users also to come up with. Now coming on to the second part, why Agilent was slow. So with respect to compliance, there were some gaps initially, which we were able to understand that, yes, the requirements of bioanalysis is specifically with respect to 21 CFR Part 11 compliance is something which is of the prime importance for this industry. And with the new version, what we have come up a year and a half back, we have tried to address probably all of those gaps. And I can say that, yes, now, not only in terms of hardware, but in terms of software compliance also, we have something very, very strong to offer this industry. So that was the two reasons, basically, what I was able to understand through my analysis in past few months, that why exactly we actually have the less share. And that does not define future, definitely, because we are very confident of what we have now as a complete portfolio. And probably in few coming years, you will see the difference.
Yeah, thanks, Dr. Thanks, Dr. Kuldeep, for sharing that insight. In the interest of time, maybe we would take up any additional questions through email. I really appreciate for sharing the technological advances, what's going in the Agilent systems, and how we can benefit from those. I really appreciate for that talk.
Thank you.
Once again, thank you, Dr. Kuldeep.
Thank you, Dr. Kuldeep, for a wonderful talk, and thank you to Agilent as well for their continual support to APA chapter for almost a decade now. So thank you for a wonderful talk, Kuldeep.
Yeah, maybe over to you, Deepak.
Thank you. Would like to thank you also, Purushottam, for a wonderful for a wonderful job you've done in moderating the session. Thank you, Purushottam. As always, you've been a very active member of our committee. So once again, thanks a lot for your support and being there. Would like to thank all the speakers also, starting from Saji Menon, Dr. Srividya, and Kuldeep Sharma for their talk and very interactive session. And if there are any questions in the Q&A, the audience can still post their questions in the Q&A tab. And if speakers are around, they can just answer them in the Q&A tab. As I can see, one question is already there for Kuldeep. And if you can just answer that on a Q&A tab, you can type the answer for them. So thank you. Thank you to all the speakers and moderators. So let's move on. So the next agenda item is going to be our recognition of sponsors. So as I said earlier also, that it is not possible to do this meeting at this level without support from all our esteemed sponsors. So a big thank you to all our sponsors, and we would like to present memento to all of them to recognize their support and contribution to APA India chapter since last decade or so. So let me start with, I would like to invite Ms. Dr. Narmada, if she is around, to say a few words or share their feedback from Natco Pharma.
Yeah, thank you. Good morning. I'm Dr. Narmada from Natco Pharma. I would like to talk about my company. We at Natco are engaged in developing, manufacturing, and marketing of drug products and active pharmaceutical ingredients in India and international markets. Natco is one of the market leader in the branded oncology medicines in India and among the country's top 3 producers of hepatitis C drugs. Our focus is primarily on niche therapeutic areas and complex pharma products. Natco has shown first of its kind in the novel drug delivery system through nanoparticle size liposomal product deoxorubicin and the generic version of long-awaited complex blockbuster drug Glatiramer in US market. The main motto of Natco is deliver affordable and life-saving drugs globally. This principle of Natco that no patient goes without medicine for want of money has set the trend of affordable pricing and patient assistance in the Indian oncology market. Natco also has a corporate social responsibility wing, the Natco Trust, that operates in Hyderabad city of state of Telangana and Guntur district of Andhra Pradesh with education support programs, health, hygiene, sanitation, livelihoods, and employment generation. Thank you all. Thank you, API.
Thank you, Dr. Narmada, and would like to thank Natco Pharma through you for their continual support to APA India chapter. Thank you, Dr. Narmada. Thanks a lot.
Thank you.
So would like to invite now Mr. Sharad from Thermo, if you are there.
Yeah, good morning all, and thanks Dr. Deepak for inviting me to speak few words about this conference. It is fair to mention that the conference was a great success and many people have contributed in so many ways to turn this event into a smoothly running meeting with a very interesting presentations and posters in these critical situations where we generally talk about to have any meetings and conferences virtually only. So most importantly, all team members made a very good atmosphere for discussion and networking with the all speakers as and when they got an opportunity to interact with them. I think it was a great platform where we build a close relation with the speakers as well and clear our doubts and queries. We look forward to having a long association with you as a mutual partner in our future success. Once again, I appreciate your and your team's hard efforts. Wish you good luck for APA 2022. Thanks.
Thank you, Sharad, and thanks for the kind words. And we've always been grateful to Thermo Fisher. I would say since last 12 years, Thermo Fisher, since the inception of APA meetings, Thermo has been with the APA India chapter. So thank you for your continued support. Thanks a lot, Sharad. And this memento, we would, this is just a virtual presentation, but we would make sure to send it across to you, the real memento as well. Thank you.
Thank you. Thank you, Doctor.
So with that, I would like to thank Thermo. Can I now request Shiva from Nanotemper?
Thanks, Deepak. Good morning to everybody. I'm Shivram, Head Business Operations for Nanotemper Technologies. To brief you, Nanotemper Technologies provides a couple of platforms for the protein characterization, particularly the protein-protein interactions and the protein stability studies. So regarding this APA, I really have some special words to thank to Deepak. It has been a wonderfully organized sessions. We had a very good interactions and we see a great audience and also very interactive speakers, which is like, a very good platform. So initially when it was virtual, we were slightly skeptical how this is going to be, but it is wonderful sessions we had. We thoroughly enjoyed a lot of knowledge transfer has been done during these sessions. And once again, thank you so much for giving us this opportunity, and we look forward for the next APA sessions. Thank you, Deepak. Thank you so much.
Thank you, Shiva. Always a pleasure, and thanks to Nanotemper for their very active participation. And since you have been associated with APA, I think every year you have been with us. So we hope to have you with us in future as well. And as we were discussing, next meeting hopefully we'll be having in person. So it would be a great pleasure to meet you also in person next year. Thank you, Shiva.
Thank you, Deepak. Thank you so much.
May I now request Ashutosh, if you are there.
Good morning, Deepak. Good morning, all the panelists and the participants. So it has, to talk about the API, we as a Shibatsu has a long association with the API, and this has been really a wonderful new normal conference, I can say, where we can get a chance to attend all the talks, no overlapping. And thank you, Deepak and the API team for such a wonderful arrangement, systematic planning. We as a Shimatsu, we all are aware that we are the leading suppliers in the analytical and measuring instruments. And we presented 2 talks in this conference, 1 poster. So if you are interested in any of these talks, maybe later on API team can share with you. So you can revert to us, you can contact us on the website, and soon we can, soon see you all in the next conference, which I hope will be a physical offline conference where we can meet, greet, and share our knowledge, experiences, and the updates. Thank you, all of you. Goodbye.
Thank you, Ashutosh, and through you, we would like to thank Shimatsu also, and just for the knowledge of the audience, I would like to state that Shimatsu has been with us almost for more than, I would say, a decade, and they are among the first sponsors who always come on board as soon as the meeting is announced, APA annual meeting is announced. So we really value your participation and support. So thank you, Ashutosh, and thank you for supporting this virtual conference. And as we were discussing, next meeting would be definitely in person, and looking forward to connect with you again. So thank you, Ashutosh.
Thank you.
Now may I request Samula, if he's there. Sam, if you can say a few words or concluding remarks from your side on behalf of Origin.
Yeah, good morning, ladies and gentlemen. It was a great pleasure to be a part of APA. From last 1 decade also, we are associated with the APA. And it was nice, as everybody said, we were thinking that it was virtual conference. However, how it will be, we were wondering the participation, how it will be. But however, it was excellent. The speakers for their excellent thought-provoking talks, and the session chairman also was well organizing the sessions and animating their sessions and keeping the things both under control and with a reasonable on time. However, the most important thing is I want to thank all of you for the participants as well as the organizing committee in terms of attending this virtual conference and making it success. So it is a great opportunity for all of us for encouraging in terms of communication between the facilities or the ideas and the common interest. So we are able to exchange these ideas and all. So in terms of Origin, we are a development stage company. We're engaged in discovery and clinical development of novel and best-in-class therapies to treat the cancer. We are a wholly subsidiary of Dr. Reddy's, and we focus on the precision oncology and oral immune checkpoints and TS17 pathways. So we have compounds at different stages, both preclinical and clinical development. So with this, I'll thank all of you for your support and fruitful discussion. A big thank you for all again.
Thank you, Samula, and to you, would like to thank Origin Discoveries Technology Limited for their support to AP India chapter, and we wish to continue this, and would also like to have you back again next year. And we had a wonderful plenary session also from Dr. Murli Ramachandra, the CEO of Origin. So it's always been a pleasure to learn about the Origins and their offerings and what they have achieved. And as you said, rightly in oncology space. So thank you and good luck to you, Samula. Would like to now invite Ravi, if Ravi Krovidi from Vida Clinical, if he's
Hi, Deepak. Good morning. Good morning, everyone. Thank you so much for the opportunity that you have given us to present in this conference. And I'm glad to see the energy levels of Deepak, the way things have been organized in terms of all the talks. I think it's an excellent knowledge sharing platform. And the topics were well chosen. Thank you so much, Deepak. And thanks to everyone here. And just a quick, most of you may be knowing what Vida does. Vida is a clinical research company. It's a pioneer in the field of clinical research, offering clinical services to various domains, both small molecules and large molecules. In terms of large molecules, we have a dedicated division, which is Ingenuity Biosciences, and I currently lead the Ingenuity Biosciences. Great, and hopefully, we'll soon have a hybrid version of this APA conference. Good luck, everyone. Have a nice day. Thank you.
Thank you, Ravi, and thank you to Vida Clinical for their support, and looking forward to connecting with you again for the next year in-person meeting. Thank you, Ravi. So may I now request Narayan, if you're around, to share your concluding remarks.
Thank you, first of all, to APA team as well as Deepak for hosting such a wonderful sessions. Earlier, we used to see 1 or 2 days webinars, but it's a long spend webinar with a lot of thought leaders from the industry and the regulator part of it. So it's a really great event. On this opportunity, I would like to talk about a few words related to USP. I'm sure most of you already aware about, but I want to say this, that I'm very proud to be a part of our organization or representing organization having more than 200 years of experience. So USP has worked to build the trust where it matters a lot. In the words medicines, dietary supplement, as well as in food ingredients. Though our rigorous science and the public quality standards we set, USP helps protected the patient safety and improve the health of people around the world. So USP is not focused on any specific country. It's a worldwide recognized, as well as having most of the countries using USP as their one of the standard setting organization in their countries. USP is independent. That's the beauty of USP. Scientific non-for-profit organization focused on building trust in the supply of safe and quality medicines. We are working to strengthen the global supply chain so that the medicines people rely on for health and available when needed and work as expected. We in USP, as you know, that we do these we give these reference standard to the pharma industry. Also, we organize a lot of education courses so that people can understand the pharmacopoeia as well as they understand the techniques related to it, and that's going to be helpful for them to work in day in, day out, work in the laboratory. So in case you have something, you can reach out to us. We will happy to support you in that area as well. With this note, I thank you to all of you for joining the session. I'm sure you are going to be overwhelmed with the knowledge received during the sessions. Thank you.
Thank you, Narayan, and thanks to USP for their support. And this is just a small memento to recognize the contribution and support from USP. And hopefully we'll connect again for the next year meeting as well. So thanks, Narayan. Thanks a lot. And just for the audience, we had at least 2 to 3 wonderful speakers from USP who shared their knowledge, and they were highly, all the talks were very well received. So thank you once again, Narayan, for coordinating all this thing, and look forward to your continual support in future as well. Thank you.
Thanks a lot.
So would like to now invite if Dr. Kuldeep is there. If Kuldeep, if you can just share your concluding remarks. from Agilent.
Yeah, thank you, Deepak. This was a really good opportunity to come to this forum and talk to APA participants in detail, to present with them about our unique upgrades which we have come up with in this workflow. And especially coming on to APA conference and the organizing committee, I would like to thank your team also, because probably this is the only conference which is organized by the scientists and is for the scientists. Usually we see conferences wherein technology vendors will take a lead and probably they will drive the agendas and the discussions. But this is a forum wherein I see more of the scientific participants actually taking a lead in deciding what topics and what discussions should be taken up. Coming on to Agilent, we focus on continuous innovation and improvements, and we look forward to see how these innovations will be supportive in the workflow optimization for the industry. Thank you again for all participants for joining, and APA committee for organizing this conference.
Thank you, Kuldeep, and thanks to Agilent once again. They have been with us for almost a decade now. Thank you to Chirag as well as Kuldeep for a wonderful talk today, and hopefully we'll have you in person delivering the talk next year when we do the physical meeting. So thank you, Kuldeep. Thanks again. Now from In Vitro Edmet Labs, I think I got a message from Dr. Lee. So just would like to read out the message. Dr. Lee has extended his thanks and gratitude to the APA committee, organizing committee, and for inviting him for a talk. And he has mentioned that since the inception, he has been associated with APA, earlier as a speaker and later on as one of the sponsors as well. So he just mentioned that would be more than happy to continue to supporting this meeting in future as well. And he just wishes good luck to all the participants and all the offerings of IVL, especially the hepatocytes. I think they have shared their major market share in India, and they would like to further work on that, and would seek continued support from the APA participants also, and keep using the hepatocytes and other solutions offering from IVL. So on behalf of APA, I would like to thank IVL for their support, and hopefully we'll have them again back next year as well. From SciEx, I think unfortunately nobody is here right now, but would like to extend my gratitude and thanks to SciEx, because SciEx has also been part of APA since the inception. Almost, I think, more than 12 years now, SciEx has been with us since the inception. So we would like to extend our thanks to SciEx, and we wish to have their support in future as well. So big thank you to SciEx, and this is just a memento for them, and we will send it across to SciEx after the meeting. Same way, we would like to extend our gratitude and thanks to Phenomenix, one of our sponsors who are always there along with SciEx to support APA India chapter. So big thank you to Phenomenix as well. From Bristol Myers Squibb, would request if we have Prashant or Prakash, if you would like to share the feedback or a concluding remark on behalf of Bristol Myers Squibb. Prashant, Kule or Prakash Vachaspati, if you're there.
Yeah, Deepak Prashant here.
Yes, Prashant, please proceed.
Yeah, on behalf of BBRC, BMS, we'd like to thank to the conference, and this was one of the avenues where many of our participants from BBRC could get a wide variety of topics and interactions. So thank you, Deepak.
Thank you, Prakash, Prashant, and thanks to BMS. They have also been supporting. In fact, would like to state this while the audience should also know that actually the inception of APA started from BBRC, from Biocon BMS Research Center, almost 12 years ago, 2009. So we are so very thankful to BMS for their continual support. And since then, they've been supporting AP India meeting. And we really value that contribution and support from BMS and hope to have that in future as well. Thank you. Thank you, Prashant, Prakash, and all the participants from BMS and to the BMS management as well. Thank you.
Certainly, Deepak. Thanks.
Again, Sinjin, I think if Purushottam is there, maybe Purushottam, if you would like to say a few words as a concluding remarks on behalf of Sinjin.
Deepak, very short. Really thanks for the APA committee organizing members and all, really making these events very successful. And really achieving the objective of sharing and learning, and that's where the objective what organization to share what the recent advances within industries and getting it to the academy and industry partners. I would say it's a really great efforts being taken, and really appreciate and thank you for all the the APA committee members as well as the organizing committee for these sessions.
Thank you. Thank you, Purushottam, and big thank you to Sinjin for their continual support to AP India. So with that, let me move on to, so this year onwards, Wuxi AppTech have also come along with APA, and we would like to extend our thanks and gratitude to Wuxi for their support, and hopefully for coming years also, Wuxi will be supporting AP India chapter. So big thank you to Wuxi as well. And last but not the least, would like to thank ZEFSI. And ZEFSI, I would say, since they have started their association a few years ago with our meeting in Goa, I think since then they have been supporting AP India for all these years. So a big thank you to ZEFSI, represented by Rohit Gujar, and we really value your support. So thank you to ZEFSI as well. So with that, I think we have one last, but not the least, Laurus Infosystems. They are also the new entrants because, and it was at a very right moment because of COVID, when the whole world has moved to digitalization. Laurus Infosystems actually came up for our support, and with their offerings in life sciences with all the digital solutions, especially for pharma sector. So we thank you, Laurus Infosystems also, which is represented by their CEO, Mr. K.K. Bhagchandani, for their support, and hopefully in future also they'll keep supporting APA meetings. So thank you to Laurus system as well, and Zoetis. They have been big supporter of APA since quite many years, and we have a lot of participants also every year being nominated from Zoetis to attend APA meetings. So a big thank you to Zoetis for their support, and we really value their contribution and support to APA India chapter. So with that, we come to the end of recognition of sponsors session. And now I think it's interestingly, we would like to move on to our next session activity, which is the concluding remarks from the chair and the chair elects, because that's an official activity which we do always every year. So essentially the chair elect of this year, 2021, who are going to be the chair next year, they will be sharing their concluding remarks. So would like to request, start first with Ravi Trivedi from Jubilant Biosys. If you are there, you can share your concluding remarks. Would like to invite you for that. The chair-elect, Ravi Trivedi.
Sure, Deepak. So thank you, Deepak, for kind introduction. So great, like as I said, culmination today of this long marathon series of our APA India chapter, like 6 months untiring effort by all the people. So first, obviously, thanks to all the committee members, I will say, because in the background, they have spent hours to bring it into the live form, agreeing like for the Saturdays, sacrificing all the weekends, And then obviously thankful to all the speakers like across world, whichever time zone. Sometime people are like extending up to midnight to like deliver excellent talk for this year's session for evolving technology and emerging challenges in drug discovery and development. So I think all the like sessions, be it our regulated bioanalysis, discovery, biotransformation, or let's say our CMC, all are received so well and thoroughly enjoyed, all together like a different kind of experience. So thanks to all the speakers. And again, most importantly, as Deepak already said, thanks to all our sponsors accepting this virtual platform and still continuing our long association. So without their support, again, it was not possible. And finally, last but not the least, all the participants like coming on like every second, third, and fourth Saturday, attending these long sessions and making it another successful year. So APA India, as Deepak has shared, and we all know, the sole motive of this non-profit organization is to spread all the best scientific practices which are established elsewhere in the lab to established among Indian scientific fraternity. So that is the sole motive. And I think everybody supported it so well, like throughout these 12 years of the journey. And hopefully next year we will be having it again in the physical form, and Deepak and Boston Society will be announcing venue at right time. So not taking more time, and if I missed anyone, please excuse me. But thanks one and all, and for this year's successful culmination today, and wish the very best for next year, and would request to spread a word about this conference for next year session to whomever you know, and looking forward to have and receive everyone next year. Thanks once again for all the committee members.
Thank you, Dr. Ravi, and for your kind words. And would like to especially thank you also for your very active participation. And just for the understanding of the audience, Ravi has been with the APA since the inception of this meeting, so almost over a decade. So a big thank you to Dr. Ravi, the chair-elect from Jubilant Diocese. Thank you. May I now request Chair-elect Regulated Bioanalysis, Dr. Shefali Gupta from Clientha for her concluding remarks.
Seconds I am taking, and I just want to thank all backend team which is working with Deepak, to whom we never see, for successfully organizing all these virtual conferences. Go ahead, Deepak. I stop
Thank
here.
you. And when we do the in-person meeting next year, hopefully you'll be able to see all of them. So thank you, Dr. Ravi. Yeah. So may I request Dr. Shefali if for your concluding remarks?
Yeah, thank you, Deepak, and good morning, everyone. This lovely Saturday morning, I would be reverberating, all those sentiments that were shared by Dr. Ravi just now. And it has been an amazing journey spanning over these last 4 to 6 months, wherein we had these very informative sessions on bioanalytics. Initially, when we started with, it had been challenging to understand, how we would go about it, since we only had experience with in-person meetings. But with all these efforts that went in backhand under, Dr. Deepak and all the committee members who worked in pretty late hours and long hours, that these online sessions became so beautifully organized. And but it is also a very special thanks to all the speakers, to all the colleagues in the bioanalytical faculty, especially to our speakers and colleagues in the West, who had to actually wake up pretty late in the night just so that they were able to enlighten us with their experiences and insights in the entire journey of drug discovery and drug development. So all in all, an amazing experience. A big thank you to the participants who were there actively participating, contributing, and asking questions for all these weekends. We look forward to meeting all of you in person in the conference next year, APA, which would probably happen somewhere around the same time. And we look for an announcement on the details of the venue very soon. Thank you for the active participation, and I look forward to being there too. Thank you.
Thank you, Dr. Shefali. Thanks a lot for your concluding remarks. May I now request Dr. Jignesh Kotecha from Torrent Discovery, who is Discovery Chair-elect, for his concluding remarks.
Thank you, Deepak, and very good morning to all of you. On behalf of Discovery Bioanalysis Organizing Committee, comprises of Ravi Trivedi from Jubilant Biosys, Jonathan Joseph from Gentech, Prashant Kole from BBRC, Vijay Raina from Nectar Therapeutics, Bhavesh Patel from Eurofins Advinus India Limited, Ravi Shankar from Sun Pharma, and Shrikant from Lexi. We thank you all the participants, speakers, and panelists for encouraging participation in this web series 2021 of APA India chapter. We witnessed good number of participation throughout this web series, even in the weekends. So this shows faith and confidence in APA India chapter. And this increases our responsibility to make agenda up to date and bringing current affairs in these fields to the forum. And we assure you for this and looking forward to meeting you in person in next chapter of APA India 2022. Our special thanks to Deepak and Backdoor teams because we could make an excuse not attending some of the days of this web series, but he and his team didn't make any excuse, and they hosted entire chapter of this web series very effectively and efficiently. So special thanks to Deepak and his Backdoor teams. So with this, I would like to end my concluding remarks. Thank you, everyone.
Thank you, Dr. Jignesh. Really appreciate your kind words and your feedback. Thank you. And now let me move on to Biotransformation Chair-elect Samuel from Origin Discovery Technologies for his concluding remarks.
Thank you, Deepak. Good morning, everyone. It was a great and a very long journey, but it was very successful and it was very interesting sessions we had. On behalf of our Origin committee from the biotransformation side, thank you for delivering such important talks and a lot of materials so that all of us can deepen into our knowledge in the area of new technologies in both in drug discovery and development. I believe that we had learned a lot of these things in the area of biotransformation from the keynote sessions delivered by amazing keynote speakers as well as the workshop sessions. Whatever we had virtually, which are delivered by interactive instructors and invited speakers, and which is very interesting for us. Furthermore, I would like to thank all of the presenters and participants who have joined from last 6 months, about 18 webinars from all across the world. I sincerely hope that your presentations and healthy discussions and interactive sessions, all of these contributes our challenges in the drug discovery and development in our labs. And this has been during the pandemic situations, it was very difficult. We have done the whole organic committee without tireless job and during the weekends. So even though the contribution was very small, but I would believe that still it is valuable and deeper insights. With this note, thanks again for all of you. Good luck for all of you, all the participants, and looking forward to meet you in person in next APA conference.
Thank you. Thank you, Samuel. Thanks a lot for your concluding remarks, and very well said. I mean, I cannot agree more than this, that this has been a real tiring sessions of more than 6 months, and the credit goes to all the organizing committee members, the chair-elects, and everyone has done a great job. A big thank you to our audience also for being there for every webinar. So thanks, Sabila, for your comments. Now may I request CMC Chair-elect, if Purushottam is there, if he's not around, then would request Hemant, or Purushottam is there, then please go ahead.
Yeah, Deepak, really appreciate and thanks for the APA committee first, because driving such a long exercise in actually a current pandemic situation is not so easy, and really making such programs successful is really a next achievement, what I would say. Most of the things my fellow colleagues has already talk on, but I would like to really put one point from this conference perspective is really is making a way towards how do we really share the technological advances happening over the world to our fellow colleagues, and especially also bringing down those technical know-how in the industry and academia is really a key focus of this conference, and really happy to see that it's been achieved through various topics, various new advances, with different new avenues and other aspects. And also, I'd like to thank for all the speakers really taking their time out and sharing their experiences, learnings, and knowledge to all of us. Not but the least, I would also like to thank for all the participants really making this event successful. And we look forward to meeting all of you in the next year event in person. And please do share with any of a new area you would like us to cover in those meetings, and we will be happy to really work towards that. Thank you.
Thank you, Dr. Purushottam, for your concluding remarks. And I would just say that your last point is very important, that we seek feedback from the audience, like what they would like to hear, what they would like to learn. That you should definitely share going forward. We will come back with a survey also, so that it will help us design agenda for the next year in-person meeting. So thank you to all the chair and chair-elects for their valuable feedback and concluding remarks. We will proceed further with, in interest of time, I will quickly invite Dr. Vijay Rana from Nectar to share his views.
Deepak, thank you very much. Am I audible? My
Yes,
video
yes,
is not. Yeah, thank you. Thank you all. I'm actually, I requested Deepak to give me 5 minutes for this session to add my concluding remarks to what people already have said.
So Dr. Vijay, just I'm so sorry to do this, but if you can be little more, I mean, unfortunately we are running behind, so you may not have 5 minutes, but whatever time you can share. Thank you.
Sure. So I'm just going back. I just want to thank Deepak's team, organizing committee, especially Deepak, because of this wonderful planning and execution. So I will quickly go, because when people look for the solutions, we bring right kind of people, like-minded people together on one platform. If we go back to COVID-19 situation, when we had the session going on in Ahmedabad, I think 2020, and then we didn't know that this COVID-19 is knocking at our door, and we don't have any idea what to do with this. Doctors were struggling to bring the regimen therapies to prevent people from dying, and all. Health workers were risking their lives in the hospitals to save patients. But 18 months down the line, we have 9 vaccines and a pill, Merck pill, in the market now, and our mortality rates across the globe has gone down. So what I mean to say is that people have the like-minded people came together on a platform and brought these vaccines and pills to the market for the patients. Likewise, I'm thinking as a parallel, APA is also the same non-profit, as Ravi mentioned, non-profit organization, which brings people together, which is organizing committee, speakers, participants, and sponsors. So organizing committee has a wonderful planning and execution to conduct this program in a virtual manner. While as speakers, as Ravi mentioned, that they stayed late night, especially our people who delivered the lectures across the globe, and the participants who got the knowledge, and then they take back the knowledge to their labs to do to bring solutions to their individual problems, and the sponsors are bringing technologies by which we enhance our knowledge about the instrument, about the technology, about the preparation, etc., etc. So I think this is the very perfect platform for us to progress in the scientific field, which is pharmaceutical or biotechnology. So I'm looking forward for this session in the more in a in-person fashion next year, and I wish all the people best. Let us take this knowledge and spread this knowledge. Knowledge sharing is the greatest wonder in this world. And then I also, in the end, I wish all the people a happy Christmas and a happy New Year for the new year to come. And then I wish all the best to everyone. Thank you, Deepak.
Thank you, Dr. Vijay, and it's always been a pleasure to hear your views. And just for the audience understanding, Dr. Vijay has also been with APA since the inception in 2009, when the APA India chapter started, and he has been a visionary, and his contribution to the committee and to the complete growth of APA is highly recognized. So thank you, Dr. Vijay, once again, and look forward to connecting with you sometime soon again. So may I now request Prakash Vachaspati from BBRC to share his views.
Yeah, thank you. Thank you, Deepak, and good morning, everyone. And as everybody said, I think it was a great conference, 6 months long, and all of us had a lot of learning to do. And we also got to multiple speakers from multiple time zones to come and talk to us. And one of the best thing about this kind of format was that we actually could hear all the 3 sessions parallelly. I mean, if that was not possible in a live offline meeting. So from that point of view, I think our knowledge also improved a lot by listening to other areas. Otherwise, we would have been sitting in only one session. So I think I hope all the audience, all the participants also gained a lot of knowledge by this format of the conference. And I think I was just looking through it, and I think we had more speakers this time than we would have if we were actually on face-to-face conference. So from that perspective also, I think this conference was a great success, and I looking forward to meeting you all in person next year. Thank you.
Thank you, Prakash, for your valuable feedback and sharing the views. And very rightly said that we had more speakers than we used to have in-person meetings. So as we all know that we were a little skeptical whether we should go for such a long format of virtual webinar series or not. But at the end, I think seeing the response and I would say that the success is truly, it goes to you guys, for the organizing committee members, especially the audience also. So a big thank you to everyone for making this effort, learning effort and educational effort successful. So may I now quickly invite Prashant. Prashant, if you are there for your views or concluding remarks from your side.
Yeah. Thank you, Deepak, and thank you, organizing committee members and everyone who was involved in organizing this event. One thing, I think most of my thoughts are very well covered by the other committee members and everyone else. Couple of things. One was the continuity. When we always schedule or organize such conference, we make a lot of effort. And the pandemic made a challenge whether we could actually organize. And this alternative format, as Prakash and Vijay said, was an opportunity to keep in touch with our scientific community. Secondly, there were many topics which were very relevant for that time of the pandemic. And I still remember many of such talks, including the virus particles, for example, which was quite interesting to many of us in order to understand what our scientific community is taking as a challenge and what could be delivered. So I think this conference had a variety of technical, scientific, as well as management issues, and it actually catered to the large audience. So I again congratulate all the organizers and the participants, and we look forward to have in-person participation. Thank you.
Thank you. Thank you, Prashant, for your very valuable views. And I truly agree with every point you said. May I now quickly invite Ravi Shah from NIPER, if you are there for your concluding remarks and sharing some views.
Thanks, Deepak. If I can say a few words considering the time and other things, but it's really a good amalgamation of various talks. And many of you might be knowing that it was divided in a different week. So it was not like the physical event where you need to choose which talk you will be selecting. In this case, it was really good that people can choose what talk they would like to hear and others. Similarly, it was actually a dilemma when the exercise got started, how we will be doing in COVID situation, and how the response will be coming from the participants, how the questions can be asked effectively, and how it could have been answered effectively. So amazing orienting the people and orientations, as well as making this success. A big congrats to all APA members. Thanks a lot.
Thank you, Ravi. Thank you for being a very active committee member on CMC, and thanks a lot for your concluding remarks. May I now invite Hemant Bhutani from Novartis for his concluding remarks.
I think pretty much everything has been said, Deepak. In the interest of time, one thing I want to remind all of us that this is probably the only conference in the country where you can see wealth of experience either in the scientific committee or in the list of speakers. I mean, knowing these three committees, you have got at least 200 plus of years of experience on each committee. Pretty experienced professionals who are really dedicated to this cause. And you look at the list of speakers, we have been really fortunate to continue to attract the best speakers in their areas. So I think we should continue this because this excellence in terms of speakers and the committee members probably is the backbone of the success, built upon the very strong foundation which Deepak has brought through APA. So my best wishes to all the committee members and APA for a really long journey in this noble cause. Thank you.
Thank you, Hemant, for your remarks and views. Thanks a lot. Now, I think Srinivas had some meetings, so he had to rush through. So in the interest of time, we'll just move on. And before we actually we start announcing the poster winners, I think this is the right time to invite the poster judges to share just few words from their side about their experience of judging those posters, and any feedback they have, or any words of appreciation or they would like to share with the participants. So may I request Amarnath to share his views on the poster presentation session.
Thank you, Deepak. I would not take more than a minute's time. And first of all, thank you and the congratulate all the participants who has actively participated into the poster presentation. As we know that we have certain limitations on awarding the only a few numbers. There may be a couple of people who could not win the prize, but nevertheless, they also did whatever the best they can do it. I mean, from the academic perspective, I would really they appreciate all the participants because their active participations, their oral presentation, and the way they have presented and narrated their all the work, it was really appreciable. And I would really appreciate those. I mean, this is not the end for those who has not won the prize, but they should still continue their learning and the encourage to participate in the forthcoming the other webinars, and even in the next year, since we are planning to have the in-persons the conference, probably the newcomers would also be encouraged. This would also spread to the their student, that they should come forward and they actively participate into such kind of the conferences. So I would just conclude here and look forward to see that who are the winners today.
Thank you, Amarnath. And very quickly, last but not the least, I would like to invite another judge. I think a couple of them, Shrikant Dittakavi, to also share his viewpoint as one of the judges on the panel. Shrikant, if you're there.
Yes, Deepak. Yes. I want to congratulate whoever the poster winners are there. Means, before congratulating, I want to appreciate all the poster presenters. They did a very good job. As Amarnath mentioned, we have a limitation to judge the things, but every poster is carrying some or other good information to all the audience. Thank you. Thanks a lot.
Thank you, Shrikant. And very quickly, if we have Ravi Shankar from the judges panel, if he's there on the call, maybe if he would like to share a few words. Okay, so I think Ravi Shankar is all right. So now we can see you, Shrikant. Earlier we only had your audio. So great to see you, Shrikant. Thanks for your concluding remarks. And I think just a small change we have made in the agenda. That is now, rather than announcing the poster award straight away, I think this is the right time and opportune time for me to invite Dr. Professor Saranjit Singh. Sir, we are really honored to have you with us, all of you. I think he needs no introduction. All of us know him well, and many of us have studied under him, or we have learned from his rich experience. So I think he needs no introduction, but would like to request and then to have honor to hear from you, sir, to few words from you for encouragement for the participants as well as for the APA audience, sir. Sir, your line is muted. You are on mute, sir.
Okay.
Yes, sir.
Thanks very much, Deepak, for inviting me to this panel and for my presence today. I owe to my students. At the outset, my greetings to all present today at this concluding session of what I could hear very successful AP India 2021 CMC webinar series. Practically, I will say that I'm very fortunate to be associated with the APA India chapter. I have been invited to share my knowledge at times. And again, my thanks to Dr. Deepak and Boston International Conferences. My special thanks also due to because they have been patronizing my students. I can see Dr. Hemant Bhutani, Dr. Ravisha here on the panel also. And they have been also been invited to speak at this prestigious annual event from time to time. My appreciation to you, Deepak, and the Boston Society for agreeing to the request of my students in their interest of instituting an award on my name for the best poster from academia. Thank judges for doing the wonderful job. And this award will be presented, as I'm told, that it will be presented annually. Actually, the setting up of annual awards on my name was among many surprises the students gave me at my superannuation last month, at the end of last month from Naipur Mohali. I will say it is a real tribute to a living person. My salute to all my students for that. Before you announce the awards, my personal congratulations to this year award winners. Yes, I have one expectation from them, that they continue to do their best effort. It is not for only sake of themselves, but also enhancing the prestige of their families, the institutions they have been researching in and they belong to, and also to take forward the country, our great nation. Thanks again. My best wishes to all, and Jai Hind. Thank you very much.
Thank you, sir. Thanks a lot. It's always been a pleasure to hear from you, sir. And in fact, Professor Saranjit sir has retired in October, or I would say officially, that's the word we can use. But I know, sir, you are always going to be active, and we will not allow you to rest, and then we'll keep you engaged, sir, with one or the other activities going on, and we would need your seek your guidance all throughout, sir. So thank you, sir, for being here and sharing your views. And I think audience already know that this year onwards, we have started in honor of Dr. Saranjit sir, one award annually on name of Dr. Saranjit, Professor Saranjit Singh sir. It will be given annually, sir. And as long as APA meetings will happen, this award will always be a part of it, and even beyond that, sir. So thank you, sir. So now I think now is the right time to quickly move on to our next most awaited agenda item. I think everybody is eagerly waiting to look for the prizes, the poster winner. So I think we will get started with, so as you know, let me just quickly pull out the slide. I can maybe. I will just read out the on the agenda. Okay. So if any of the panelists can confirm if my screen is visible.
Yes, it is.
Okay, yeah. So just very quickly, the actual final poster session happened on 13th of November. And before that, we had received around 45 poster abstracts. Okay. And out of that, after initial screening, 15 posters were shortlisted for the final poster session. And that happened on 13th of November. So it was a 4.5 to 5 hours long live session. Again, we just thought that it should be a live session rather than asking for a recorded presentations. Live session always helps. So I'm so thankful to all the judges actually for sparing their valuable time, almost close to 5 hours on 13th of November. And also would like to congratulate and thank all the poster presenters who actually presented their work and I think actively participated in that. So we have actually selected 9 prizes out of that. So for each level, so for third prize, there are going to be 3 best poster winners, for second prize and for first prize. So as you can see, there are some cash rewards in a memento would be given to all of them. And as just now we have announced, Professor Saranjit Singh Award we have launched from this year. So this award will go to one poster presenter whose academic research, the presentation would be really shortlisted by the panelists, the judges. He will get a trophy, memento, certificate, everything. But apart from that, he will have a free registration for the next year in-person meeting, and all his travel and accommodation, all those things would be taken up by the Boston International. And this is the award which will be there every year starting this year. So this is the premises, and I think without further ado, let me just begin very quickly with I will just one by one, we'll call out the, we'll start from the poster number 9, I guess, and then we will move on further from there. All right, so I hope my screen is visible. So the number 9 is Shikha Patel. So Shikha Patel got the third prize. So she is the third prize winner. And let me quickly. Okay, so if Shikha is there, maybe Shikha, you can switch on your video if you are there. I can see Shikha is there, and Shikha, actually, you have been promoted as a panelist, so you just have to click that button, so you become a panelist, and you will be switch on your video.
Looks like you don't have an access to open the video.
Yeah, that's what I feel. So just 1 second, let me just see. I think she should be able to, because she is already a panelist. Shikha, yes, I can see you as a panelist. If you can switch on your video.
Deepak, just check her message. She said she is unable to switch on. But yeah, what you are suggesting, if she can join as a panelist, maybe she is able
No, she
to.
she has. I mean, yeah, Ravi. So she is already there as a panelist. I can see there.
Just see her message in our chat.
There's some
box.
problem in her network. Okay, all right. So I think there is some network issue at her end. So no worries. Nevertheless, congratulations to you, Shikha, and we really appreciate your participation and winning this prestigious third prize. So Shikha actually presented her work on force degradation strategies for peptides, a review from regulatory chemistry and formulation standpoint. And she also shared some of her work also with that. So a big congratulations to you, Shikha. And on behalf of APA Organizing Committee also, a big congratulations to you. So we'll move on, and would also like to congratulate for the third prize. The second prize under third prize winner is Arpit Bana for presenting the poster entitled Charge Heterogeneity Analysis of Selected Biosimilar Formulations Using Imaged Capillary Isoelectric Focusing Technique. So a big congratulations to you, Arpit. And
thank you, sir. Thank you so much.
So in fact, what we'll do is, in interest of time, I will just announce all the names except for the one which would be the first winner, and then would request all of you to switch on your videos. So Professor Saranjit Singh sir would also be there, and we'll have a spotlight on all of you, and we can have that snapshot taken. So congratulations to you, Arpit, for being a third prize winner at poster contest under APA India. So the last person winning the third prize would be Arnab Sinha Roy. So congratulations, Arnab, for winning the third prize in the poster session. And Arnab actually presented validation of a flow cytometry based whole blood natural killer NK cell cytotoxicity assay as immunity marker. So Arnab is from Clienta, and congratulations, Arnab. If you are there, you can switch on your audio and you can share your view, or else at the end, we will anyway have your videos displayed. So you get this third prize trophy also, and a certificate of appreciation. So congratulations to you, Arnab. So moving on, now we have 6 more winners. So for second prize also, there'll be 3 winners, starting from Navya Ajit Kumar Bhaskaran. So congratulations, Navya, for being a second prize winner. And Navya presented a poster on RP, reverse phase HPLC method development and validation for rapid and simultaneous estimation of hydrophilic and lipophilic drug encapsulated in a nanoformulation. So congratulations to you, Navya, Navya, for being the second prize winner. Moving on, would also like to congratulate Gantasala Subramanya Samir Kumar from USP for presenting a poster on the multi-attribute method for biotherapeutics. So congratulations to you, Samir. And if you're there, maybe you can say a few words, or maybe at the end, we can invite you to share your views. So moving on, the last winner in the second prize category is Shivani Tang for presenting. She presented a poster on phytochemical rich polyherbal preparation and exploring its efficacy in translational medicine. So congratulations to you, Shivani. You will get a trophy and a certificate of appreciation.
Thank you. Thank you so much, sir. Thank you.
Thank you, Shivani. And then be there at the end. We would request you to switch on your videos as well. So thank you, Shivani. Good luck. So with that, we come to the end of second prize announcements as well. So finally, now we are getting into the category of first prize winners. So we'll have 3 first prize winners, and let us just have their names. So the first prize winner, we have Aditya Narvekar for presenting a poster on pH-dependent aggregation and conformation changes of rituximab using SAXS and its comparison with the standard regulatory approach of biophysical characterization. So many congratulations to you, Aditya. You get a trophy and a certificate of appreciation and other prizes which are there for the first prize winner. So congratulations to you, Aditya.
Thank you, sir.
And then let's moving on. We would also like to congratulate Marianne Saldahana for first prize winner, and she presented her work on extracellular metabolite profiling by LCMS for bioprocess optimization. So a big congratulations to you, Marianne, for your good research work and a good poster presentation. Congratulations.
Thank you, sir.
So with that, we come to this one last prize, the first prize. I'll just hold up on that because this is going to be the same. The committee has decided the same person will also get Professor Saranjit Singh Memor Award as well. So this is the person who will get the first prize, who happens to get the first prize from academia. He is the first prize winner for the posters contest and also will be awarded Professor Saranjit Singh Memor Award. So since it is virtual, I can't ask any guesswork because I think it's not possible to do it. So let me straight away go ahead and announce the name. So I go back here and this is the name. So many congratulations to Ditiksha Desai. She is the first prize winner for APA India 2021, and she presented a poster on cyclosporine delivery from graphene oxide laden contact lenses for management of dry eye syndrome in vitro and in vivo studies. So congratulations, Ditiksha, for a great work you have done and a very nice presentation. And committee and the judges were together unanimously. They were convinced that Ditiksha should be the first. She will be the first winner, but she should also get the Professor Saranjit Singh Memor Award as well. So many congratulations to you, Ditiksha.
Thank you, sir.
And maybe before I invite all of you to just switch on your videos. Let me quickly open the Professor Saranjit Singh Memor Award as well. So Professor Saranjit sir, this is the Memor Award which will be awarded to Ditiksha Desai for research work on cyclosporine delivery from graphene oxide laden contact lenses for management of dry eye syndrome in vitro and in vivo studies. So in fact, if it was an in-person meeting, I think sir would have handed over this this memento in person to the winner. But since it's a virtual, can't help it. So this is how we have to offer it to them. So just few words from you, Professor Saranjit sir.
Yeah, extremely happy to see the quality of the posters that have been presented or the research work that has been presented, although I was not evident to all those discussions that have happened. But my congratulations to all of them. And you are all the winners. And of course, some selection has to be done for the top person to be given the award. And very happy that Dikshita has won this one award that has been instituted by my students on my name. So it would be happy situation. I would have been happy that if the Dikshita comes online and tells about where the work has been done, which institute the person belongs to, And in future, I will request Deepak that all the certificates should carry the institution name also, so that they can, the institute also name goes on with the certificate. So thank you very much, Dikshita. Again, congratulations, tons of congratulations to you for doing excellent work. Thank you so much.
Thank you, sir. Thanks a lot for your encouraging words.
I'm honored to bestow this award to you.
Thank you, sir, for your encouraging words and your feedback taken, sir, suggestion on putting the institution name as well. So may I request Ditiksha, if she's there, to share reviews or what she's feeling right now, and at least as Dr. Saab mentioned, please share your background and about little bit about your research work and where did you carry out that work, if you can share. Ditiksha, if you're there. Okay, so while okay. Yes,
Good
we
morning,
can hear
sir.
you, Ditiksha. Yeah.
Yeah. Am I audible, sir?
Yes, very much.
Yeah. Okay, sir. Thank you so much, sir, for giving me such an prestigious award. Actually, my work is on ocular drug delivery system and to use a novel delivery system, that is contact lens. So a lot of research has been going on this contact lenses, but now this is coming to the reality. We have one lenses in the market also, which is doing with the drug delivery. So this will become the future. It will bring a paradigm shift in the ocular drug delivery system, and we are working on the same. At my institute, that is Maliba Pharmacy College, we are also doing some contract research work with one of the companies, and we are helping them to doing R&D with several molecules along with what I have done. So this was my PhD work, and that's it. I'm very happy with this award. Actually, in this conference, I wanted to share one thing. All the posters were of different category. I think drug delivery was only that of mine. The rest all was on chemistry and analytical part. So I was a little bit thinking that this will be applicable in this with the award.
Thank you,
That's it from my side.
thank you, Ditiksha, and once again, many congratulations to you. And very quickly, since Professor Saranjit sir is also there in the frame, if I may request all the poster winners, all the poster prize winners, if you all can switch on your video, we can have quickly one snapshot where everybody will be in the frame. Okay, so we have Aditya already. Can I request Marianne and Shivani and everyone who is present right now, if you are in a position to switch on your video. Yeah, there we see Marianne. Good to see all of you in your lab. So great. So the work goes on. Shikha is also there. Can I request who else is there? Yes, we have Navya Bhaskar also, and Arpit is also there. Arnav is also there. Okay, let me just put it here. Great. So, and Samir has also joined. So, I think we have almost everyone except for one or two, I guess. Except for one person, I think everybody else is here. So anyway, nevertheless, really appreciate you, all of you, and big congratulations to all of you for doing such a great work, and keep doing good work and good research. I think you are the future of our country, and so glad that all of you have blessings of Professor Saranjit sir, so that you guys do a great. So let's have a word virtually, but let's have a big round of applause for all the poster session winners. Okay, so very quickly, I don't want to take you on spot, but just one sentence, if you guys would like to share. We have that much time, and then we move on quickly. So starting, Ditiksha has already shared her views, but just one or two sentences you can talk, and then we can invite others also. Over to you, Ditiksha.
As I told you, sir, such events like international conferences, webinars, we get a lot of things to learn. It gives us new perspective to carry out new research and to work in the respective fields. So thank you so much.
Thank you. Thank you, Ditiksha. May I request Aditya?
At the outset, I would first like to thank the organizers for providing this opportunity for individuals from academia to present at a venue where we have a input from the industrialists as well as academicians. So this makes the APA India conference very unique, and thanks to all the organizers for making it happen. Thank you very much.
Thank you. Thank you, Aditya. May I request Marianne to say a few words?
Yeah, good morning, everyone. Thank you very much to Deepak sir and all the organizers for giving us this platform to present our work. For us being PhD graduates, this is actually a very good platform for exposure and to show the world the work that we are working on. So thank you so much for giving us this opportunity. And it was a great pleasure being part of this conference. Thank you.
Thank you, Marianne. May I request Shikha?
Yeah, am I audible?
Yes, Shikha, please proceed.
Yeah, thank you so much to the APA India 2021. I'm really feel very happy after a very long time of my maternity. In spite of being mother of 5-year-old kid, after a long time I have presented something. I have started my PhD and I have won this award. So thank you so much, APA India 2021 and Mr. Deepak. I think I was the last one who have enrolled in this poster session, but still have done well. So I'm feeling really very motivated and really want to be there in the next part also. Thank you.
Thank you, Shikha, and congratulations to you. May I request Navya?
Good morning, sir. Am I audible?
Yes, Navya, please proceed.
So I would like to take an opportunity to thank one and all who have been a part of this APA conference. And my heartfelt thank you to you, sir, for considering my poster, even though I was a little late. This is the second time I am participating in the APA conferences. The first time I had attended when it was carried out internationally at Boston. And it was also during the tenure of my PhD only. I am really thankful that I get this platform to present my work. I thank everyone for giving me this opportunity. Thank you.
Thank you, Navya. And may I now invite Arpit and would like to share this with everyone that Arpit was the first poster winner last year. So congratulations to you, Arpit. You have been consistently doing good, and glad that this year also you could make it. So congratulations to you. Over to you, Arpit.
Thank you, sir. Am I audible to everyone?
Yes, sir. Please proceed.
I would like to thank Deepak sir as well as all APA platform for giving me the opportunity to provide present my poster, both the previous year today and this year also. And I would also like to thank all the judges for recognizing my work and as giving the opportunity to present here, and also for all the presenters as well as the speakers. That it was a very good opportunity as well as a very good you can say experience to gain all this experience from all of you. As someone said that more than 200 years of experience for each and every theme. So it was very good experience for me, and I would like to thank everyone for giving me this opportunity as well as for giving me the award. Thank you so much.
Thank you, Arpit. May I request Arnab to share your views?
Yeah, am I audible?
Yes, Arnab.
Yeah, thank you, sir. And thanks APA members and organizing committee for this wonderful platform. The entire session was really helpful. And I would also like to thank my organization, Clianta Research, for giving me this opportunity to showcase my and my team members' work over here. And so thanks again.
Thank you. Thank you, Arnab. And last but not the least, let me request Samir to share your experience with APA and being a poster winner. So over to you, Samir.
Yeah, thank you, Deepak, and very good morning to you all. I'm very happy to participate in APA 2021. And actually earlier also I'm closely following APA conferences and all. It's really a very great and pleasure to participate always. And I'm thankful to my organizing committee side and also from my company side USP. And because we are facing kind of challenges, pandemic challenges and all this, from long marathon run like 6 months overall, making a very good sessions and very interactive and sharing knowledge. And thanks to Deepak and organizers for encouraging all the youngsters for giving the presentations. And I'm very happy to participate in future as well. And thanks to one and all. Be strong and be safe. Thank you.
Thank you, Samir. And lastly, would request Honorable Professor Saranjit sir for your final few words of encouragement and blessings for all the poster winners.
Okay, friends, as I said earlier, you are the people who have to take the country forward. Continue doing good research, publish in best of the journals so that we can, and try to build up whatever can be translational. So India has to be innovative. It has to leave its mark to the world community. At my institute, in my group, you can see some of the students who have gone to pinnacle situation or the achievement they have done worldwide. So I think we all need to be together, and APA is such a good forum for learning that I have been also participating and even listening to the speakers and the best of the world knowledge that had been shared on this forum. Again, my congratulations to you all. Do best of what you can. And thanks again to Deepak for really inviting me and making me such honored. I feel honored today. Thank you so much, everybody.
Thank you, sir. Thanks a lot. And very quickly, I think we are in last 4 to 5 minutes of our session, I guess. So let me request all the organizing committee members, if you guys can just switch on your videos, then would like to have just one together in a frame with Professor Sab, so that we can have one snapshot also taken. So would request all the panelists to please switch on your videos.
And Deepak, probably we can also clarify that, the margin between the first winner, second and third was very, very low. It was very, very close. So they should not feel that the first winner has a very high number and second is lower, but the margin between first, second and third was very, very close. So they should not feel that they are the third position, second and first, but all are the winners.
I completely agree, and a very valid point. I think I've just missed out mentioning that, but I think all the posters were really good, and it was tough time for judges to really choose 9 out of 15 posters. And again, I would say that the marking, as mentioned by Amarnath, it was so close. I think hardly 1 or 2 points difference also we have seen for few of the poster winners. So great job, all of you. And let me just quickly now take a snapshot. So I think, okay, so all of you who could switch on your videos, you can just switch on and we'll take one snapshot now. And I hope to see all these happy smiling faces again next year in person. And we would definitely beat COVID or any such viral infections, pandemics for sure by next year, and we should be able to back to our normal life and meeting in person. So thank you, and looking forward to see you all. So now I think I had taken a few snaps already, so it's done. So now let me just very quickly, in last 3 to 4 minutes, what I will do is I will just take a very quick journey. Glimpses of especially the webinar series, what we have done, because I'm sure many of you would have even forgotten like what was our first webinar, because today we are on 18th webinar. So I'll just quickly browse those slides so that all of you can just see what were the talks, what are the webinars, and then all the webinars, all the talks will be uploaded on Boston International channel as well. So later on you can view that as well, okay? And while this is happening in next 3 to 4 minutes, would request all of you on your screen, you will see this poll, the last activity for us, that is the webinar poll. I'm just launching it. You can just put up your, responses. You can submit your responses. So while this is happening, I will take you through that slide deck, okay? So I have just launched the poll. So it should be on your screen now. Okay, so let me now quickly take you through the So while you are just submitting your responses, we started our journey from 12th of June this year with a monthly theme for June was repurposed drugs, vaccine and diagnostic development to combat emergency disease outbreaks. And we had our first webinar on 12th June, Ranjan Chakravarti, Rama Rao Kalakuntla and Kapil Maithal. They were the first 3 speakers on 12th June talking about repurposed drugs, vaccines and diagnostics because we selected the topic in the monthly theme, keeping COVID into center stage. So as you would have already attended, so Ranjan Chakravarti, Rama Rao Kalakutla, and Dr. Kapil Maithal. Then second webinar was on 19th June under Discovery Biotransformation, and we had Nagendra Chimuturi and Anup Madan from US joining us, and great talks given by Nagendra on viral gene therapies, followed by Anup about dynamic monitoring of disease using genomic platforms. Third webinar was on 26th June. That was on the CMC session. And we had 3 very great talks from Hugh Hughes, Prabuddha, and Ashutosh. Hugh talked about emergency vaccines against emergency diseases in animals. Prabuddha talked about the world's first triple antigen virus-like particle against SARS-CoV virus and oral delivery. And Ashutosh talked about expanding role of mass spectrometry in vaccine analytics. The fourth webinar for the month of July, where the monthly theme was technological advancement under regulated. We had Jesse Ricks from Somru Bioscience Canada and Vinay Bethinani from Phenomenics India presenting their talks. So again, well-received talks from Jesse Ricks and Vinay, latest development in sample prep, how to achieve high sensitivities in SLE and faster SPE for bioanalytical applications. Then on the Discovery webinar 5th, we had June Q from University of New York, Sunny Buffalo, and Gian Albert from SciEx. So June gave a great talk on novel trapping micro LC FAMES SRM MS method. And Gian talked about new era of precision bioanalysis using SciEx 7500 LC-MS platform. With webinar 6th under CMC, we had 2 talks, one from Amit and then Ajit. Amit gave a great talk on transdermal drug delivery, past, present, and future. And Ajit, his talk was also well received, particle engineering for inhaled drug delivery. For the month of August, that was our plenary session. So all the talks were plenary, where we started with Girish Kapoor from USP, KK Bakchandani under regulated. So Girish talked about supporting public health responses for COVID-19. And KK Bakchandani talked about digitalization and its impact, why the world has accelerated its adoption. Under Discovery, we had 3 back-to-back great talks from Jash. He talked about next frontier in ADME science, predicting and verifying transporter-mediated drug clearance and tissue concentrations. We had a great talk from Murli Ramachandra from Origin, who talked about the changing landscape of target-based drug delivery. And then Kevin gave a wonderful talk on improving compound quality, physicochemical and DMPK properties in drug discovery. We continued the plenary session with CMC, and we had in CMC great talks from Rajiv Singh Raghuvanshi from IPC, and Debashish Banerjee from Novartis. So Dr. Rajiv talked about IPA, Indian pharmacopoeia, relevance in present context, and Debashish talked about data science and artificial intelligence. And then we moved on to the next month of September, where we talked about regulatory challenges, learnings, and opportunities. And Clarinda Islam from Somru, Amarnath from Viatris, and Mitesh from Zydus gave a wonderful talk. So Clarinda talked about overview of global regulatory landscape. Amarnath gave a talk about bioanalytical challenges in new learning, and Mitesh talked about reduction in failure rates in regulated bioanalysis, human error perspective. So with discovery and biotransformation for the monthly theme of regulated challenges, we had Krishna here and Abhay Sangamwar from NIPER giving great talks. So Krishna, as usual, his talk was well received. A brief introduction to non-linear PK and its clinical consequences, and complementary to that, Abhay talked about supersaturation behavior of drugs in gastrointestinal environment and its implication in drug development and clinical formulation development. So both the talks were well received. Then we had under CMC, novel excipients and extractables and leachables. So there we had a real interesting talks from Raj, Suryanarayana, Ravi Shah, and Mohinish Sahai and Amzad Anwari. So Raj gave a talk on excipient phase behavior in frozen and freeze-dried systems, potential implication on drug stability. Ravi gave a very interesting talk on drug excipient compatibility study, QO-WADIS. And Mohinish covered extractable and leachable studies for complex LVP drug products, technical challenges and considerations. So with that, we moved on to the month of October, where biotherapeutics was the focus area. And we had Darshna, Arjun Kompenjan, Aparna, and Jian giving very interesting talks. So Darshna gave a talk on bioanalytical strategies for biospecific antibodies during clinical development. Arjun, very nice talk on PK assay development and beyond. And Aparna talked about large molecule bioanalysis during COVID-19. And Jian talked about quantitative challenges and psych solutions. And under discovery, we had Sampada Kopkar, Sanjeeva, and Rene Vink. Sampada gave a talk on Shimadzu Aggregate Sizer, tool for evaluating biopharmaceutical aggregations. Sanjeeva gave a very interesting talk on high resolution LC-MS based proteomics. And Rene talked about the solution for today's challenging drug discovery pipeline. And finally, we had CMC concluding webinar for the month of October, where Satish from Moderna, Meendu from Thermo, and Anupal from USP gave a very thought-provoking talks. So surfactants in protein formulation by Satish. Meendu talked about state-of-art analytics for characterizing new pharmaceutical modalities, and Anu talked about ensuring quality development of monoclonal antibodies with USP standards. And finally, the current month of November, on November 13th, we had one talk, which was really interesting from Russell. And so Russell Wiener gave a very thought-provoking talk on conducting clinical trials in low and middle income countries, understanding the pre-analytic and logistic challenges. And as we have been talking, we had our most exciting event of this conference, our AP India poster session also on 13th November. So these are all the poster presenters. So in total, 17 posters were presented, out of which 9 winners today we have announced. Finally, on 20th November last week, we had Discovery Biotransformation, where very interesting talks were lined up. Albert Lee from In Vitro Med Labs, Peter from KU Leuven, Belgium, Alex Sads from WuXi, and Vikas from Vida Clinical Research. So where Dr. Albert Lee talked about 999 elite cryopreserved human hepatocytes. Ennert gave a very nice overview about membrane transporters, role in drug discovery and development. And Alex from WuXi talked about DNA encoded libraries in drug discovery. And last talk was from Vikas, who talked about Central Bioanalytical Lab, NC's clinical trials, case studies. And finally, today's webinar we came to with Saji talking about multi-parameter stability characterization, Dr. Srividya talking about PAT in crystallization process development, and Kuldeep Sharma talking about bridging technology advances with regulated bioanalysis, agile technologies. So with that, we come to the end. This was just a very quick glimpses of what we have been through. So as you can see in this last slide, hopefully, again, I have not really put it as a confirm, but a lot of feedback have come for Hyderabad for an in-person meeting venue. So would like to thank you one and all for your very active participation. Would like to take this moment to thank all the participants for your active participation. A very, very big thank you to organizers because without their support, it was just not possible to run such a long, exhaustive webinar session. And I know whatever we can do for our organizers is less, but yes, we are planning to have a separate organizer get together meet. Again, because of the travel restrictions, if everybody cannot be, it's not possible to meet at one place, maybe then at least in 2-3 cities, we'll be doing a special organizer meet where we'll actually do the complete dissection of this entire conference event. And we will really acknowledge your participation, your contribution, and we will have some quality time together. So just be prepared for that. And hopefully we will have it within December itself before Christmas. So the big thank you to all our speakers. I think many of the speakers from US and Europe who were joining at around midnight. I think some were even joined at 2 o'clock also. So I think I really appreciate. I mean, no words no words can really justify this, but I think a big thank you to all our speakers, all the attendees. Actually, honestly speaking, we were very skeptical. We were not sure about the success of this, but at least I have seen 60-70 people every Zoom webinar every Saturday attending. So I think on a lighter note, who will do that? I mean, if I was one of the attendees, I'll never attend such a long conference. So anyway, but on a serious note, I mean, a big thank you to all of you. I mean, this has been a marathon effort, and without support from the audience, from the participants of APA, it's just not possible. So a big thank you to all of you. And with that, let me now check if poll. All right, so I think most of the people have submitted their polls. So let me end the poll. So I'm going to end the poll now. Okay, so now I'm just downloading the results, which will take at least 2 minutes time because the internet thing, it takes 2 minutes anyway. So meanwhile, if anyone wants to share anything, feel free. I will just take 1.5 minutes. The sheet gets downloaded, then immediately we'll display that.
May I say something?
Yes, sir, definitely. Please proceed.
I think all of us who are present today in the audience should have a big clap and standing ovation. I would say standing ovation for you, Deepak, and the organizer for having such a. I was very thrilled to see what all talks have been delivered and what technology and all sections of science that has been discussed. So great of you and all the organizers. A big clap to you all. Thank you so much.
Thank you, sir. And wish to have your blessings and good wishes always with us. And as I said, sir, officially you would have retired, but we will definitely keep you busy coming back to you with one or the other request every now and then. So thank you. All right, so let me now stop the live stream.